Malabika, am I audible? Malabika, am I audible? Malabika, please unmute yourself and test or not.
আমাকে শোনা যাচ্ছে?
হ্যাঁ হ্যাঁ, একটু ভিডিওটা অন কর, দেখা যাচ্ছে কিনা দেখে নেবো, আমার স্ক্রিন শেয়ার করা আছে। শুধু তো ভিডিওটা একটু অন কর, দেখে নি।
You cannot start your video because the host has stopped it.
কিন্তু সেটা তো হবে না, আমি তো প্যানেলিস্ট হয়ে গেছিস তো। দাঁড়া, লেট মি সি, জাস্ট আ মিনিট। না, হওয়ার কথা না তো, রিচেক কর।
আচ্ছা দাঁড়া, স্টার্ট ভিডিও তো বারবারই।
ওকে, আমি স্টপ শেয়ার করলে দেখে নিচ্ছি, দাঁড়া। হ্যাঁ, এই তো, ইটস ওকে, ইটস ওকে, শেয়ার করা ছিল, দেখতে পাচ্ছি। হ্যাঁ, দেখতে পাচ্ছি।
সেটা তুমি আমাকে যেটা দেখছো ফোন থেকে দেখছো, নট ভায়া দ্য ল্যাপটপ।
কেন?
আমি ওই জন্য আমি
না,
ল্যাপটপ থেকে
দুটো। আগে কোনটা দিয়ে জয়েন করেছিস?
এবার দেখতে পাচ্ছো? ক্যান ইউ সি মি নাও?
না, তুই কোনটা দিয়ে আগে জয়েন করেছিস? দুটো দিয়ে জয়েন করেছিস?
ফোন দিয়ে জয়েন করেছি।
ফোন দিয়ে জয়েন করেছিস বলেই ফোন থেকে দেখতে পাচ্ছি। দাঁড়া, এবার ল্যাপটপ থেকেও জয়েন করেছিস তো? দাঁড়া, এক সেকেন্ড, এক সেকেন্ড। তাহলে আমাকে ওটাকে করতে হবে, প্যানেলিস্টে আনতে হবে। দাঁড়াও, সেম নাম, সেম নাম। ল্যাপটপে কি নাম আছে?
ল্যাপটপে তো ডক্টর মালবিকা ভট্টাচার্য আছে গো, সেমই তো।
না, দুটো দুটো আলাদা আলাদা ডিভাইস থেকে হলে দুটো ডিভাইসকেই আমাকে অ্যাক্টিভেট করতে হবে।
আচ্ছা, আমার আইডি হচ্ছে 312639।
দাঁড়া, একটা আমি করে দিয়েছি, সেটা প্রবাবলি ফোন।
তাহলে সেটাকে আমি কি লগ আউট করে যেটা থাকছে সেটাকে করে দাও তাহলে আগে।
আচ্ছা, আগে ফোনটা লগ আউট করে বেরিয়ে যা, লেট মি সি। আমি আরেকটা নাম কিন্তু দেখতে পাচ্ছি না এখানে।
ম্যাডাম দুটো নামই দেখাচ্ছে কিন্তু।
কোথায় রে?
এখন এখন একটা দেখাচ্ছে, এখন একটা দেখাচ্ছে। একটা তো উনি বেরিয়ে গেছেন, যেটা
হ্যাঁ,
কোহেস্ট ছিলেন ওটা উনি বেরিয়ে গেছেন, এবার একটা দেখাচ্ছে ডক্টর মালবিকা ভট্টাচার্য।
হ্যাঁ হ্যাঁ, এটা করি, মেক
একটা দেখাচ্ছে।
হোস্ট, ওই জন্যই গন্ডগোল হচ্ছিল তখন। আচ্ছা, আমি তখন দুটোতে করিনি, দাঁড়া।
আমি
মানবিকা, এবার দেখ, ল্যাপটপটা আগে অন কর।
হ্যাঁ, ল্যাপটপটা অন করেছি, এবার দেখি স্টার্ট ভিডিও। হ্যাঁ, এবার স্টার্ট হচ্ছে, এবার আর প্রবলেম হচ্ছে না।
দেখি, এখনো আসেনি এখানে।
হ্যাঁ, দাঁড়াও। আচ্ছা, ফোনটাকেও আমি ব্যাকআপে রাখছি।
হ্যাঁ হ্যাঁ, যদি কোনো কারণে গন্ডগোল হয় ফোন দিয়ে ওঠো, আগে এটাকে দেখে নি।
হ্যাঁ, বিকজ ইন এনি কেস এটা প্রবলেম হলে আই ক্যান টেক ওভার অন ফোন।
এবার ঠিক আছে, এবার আবার ফোন দিয়ে আলাদা করে ঢোক। ফোন দিয়ে ঢোক। মনি, সি whether you can see Malabika.
ওকে ম্যাডাম, আমি দেখে বলছি।
হ্যাঁ,
দেখা যাচ্ছে, দেখা যাচ্ছে, দেখা যাচ্ছে, দুটোই দেখা যাচ্ছে।
MNOP কোথায় দেখা যাচ্ছে? ইয়েতেই, প্যানেলিস্টে?
হ্যাঁ, প্যানেলিস্টে দেখা যাচ্ছে দুটো।
ওকে ওকে, তাহলে প্যানেলিস্টে একসাথে আছে। আচ্ছা আচ্ছা, হ্যাঁ, বুঝতে পেরেছি। একটা হয়েছে, আর একটা করে দিচ্ছি।
আচ্ছা শোনো, আমি
বোথ ডান, দুটো দিয়েই অন করতে পারবি ভিডিও।
হ্যাঁ, দুটো দিয়ে অন, দুটোই কোহেস্ট দেখাচ্ছে।
পিডিএফটা শেয়ার করছি বিকজ পিডিএফ এর আমি এটা সাইজ ইজ 8.3 MB। ওকে,
তুই শেয়ার কর, দেখে নি সব ঠিক আছে কিনা, একবার শেয়ার করে দে।
শেয়ার করছি, ওকে,
so that we can check before starting.
share screen. I'm going to the share. It's coming.
Yes, absolutely fine. Absolutely fine.
So আমি পিডিএফটা দিয়ে যাচ্ছি বিকজ সাইজ কম তো কোনো প্রবলেম হবে না তাহলে টেকনিক্যাল।
নো প্রবলেম। ওকে,
আচ্ছা, আরেকটা জিনিস একটু চেক করতে চাইছি, আমি একটু ভিডিওটা করছি, দেখো। হ্যাঁ, দাঁড়া, আমি এটা বেরোচ্ছি, এক সেকেন্ড, এটা বেরোচ্ছি।
ওই ভিডিও স্ক্রিনটা শেয়ার কর।
হ্যাঁ হ্যাঁ, করছি, দাঁড়া, আগে এটা থেকে বেরোই। স্টপ শেয়ার করি, স্টপ শেয়ার।
ইয়েস,
ভিডিও স্ক্রিনটা এবার শেয়ার করছি। দাঁড়া, ভিডিওটা খুলছি, একটু ওয়েট করতে হবে।
শেয়ার করা হয়েছে? শেয়ার
না
করা
শেয়ার
হয়নি?
করিনি, না না শেয়ার
ওকে
করিনি।
ওকে, ওটা খুললে শেয়ার করে, হ্যাঁ ঠিক আছে। হুম,
লেট মি সি, ওপেন হয়েছে।
হবে, ওটা ক্লিক করলেই হবে।
চালু হওয়া উচিত তো।
হবে হবে।
সাউন্ড শোনা যাচ্ছে?
এখনো কিছু হয়নি, শোনা যাচ্ছে, হ্যাঁ এবার আসছে, দাঁড়া। এখনো সাউন্ড আসছে না কিন্তু।
তার মানে সাউন্ডটা ধরছে না।
এই একই সমস্যা আমার কালকে হয়েছিল, যার জন্য আমি ওটাকে আবার মডিফাই করছি। না, এটা কি ইউটিউবের ভিডিও ইউটিউবে কনভার্ট করলে তবে একমাত্র পসিবল। নো, সাউন্ড ইজ নট কামিং।
সাউন্ড আসছে না।
মানে
তাহলে কি
জুমে নর্মাল ভিডিও যেগুলো সেগুলো সাপোর্ট করছে না আমি দেখছি, মানে উইন্ডোজ
আচ্ছা
মিডিয়া প্লেয়ার বা এগুলো। একমাত্র ওটাকে ইউটিউবের মধ্যে নিয়ে গিয়ে করলে, ইউটিউব চ্যানেলে, তবে সাউন্ডটা সাপোর্ট করছে।
আচ্ছা, আদারওয়াইজ উইদাউট দ্য সাউন্ড আমি
হ্যাঁ
আমি
ওকে,
পেছনে বলে দেব,
একদম
কোনো
একদম
প্রবলেম নেই।
একদম,
এই
হ্যাঁ এমনি
ভিডিওটা
পুরোটাই ঠিক আছে,
হ্যাঁ
পুরোটাই ঠিক আছে, নো
পুরোটা
প্রবলেম।
দেখা যাচ্ছে তো, সো
সব
দে উইল
সব
সি দ্য ভিডিও
সব,
আর সি বিহাইন্ড, তাহলেই
হ্যাঁ
হয়ে
হ্যাঁ,
যাবে।
একদম
ওকে
একদম,
ওকে।
ওকে ওকে। সো আমরা 150 পার্টিসিপেন্ট হলে তারপরে ফর্মালি শুরু
ঠিক
করছি, হ্যাঁ 5 মিনিটস
ঠিক
পর।
ঠিক আছে, ওকে
কারণ ওই
ওকে ওকে।
নেটের জন্য একটু সময় লাগে ওদের জয়েন
হ্যাঁ,
করতে।
আমি তাহলে এখন ভিডিও অফ করে রাখবো না থাকবে?
অ্যাজ ইউ উইশ।
থাক, একবার একবার অন হয়েছে যখন। আচ্ছা,
থাক
একটা
থাক,
জিনিস,
হ্যাঁ
বলার সময় কি অন থাকবে?
হ্যাঁ হ্যাঁ হ্যাঁ, কারণ
ওকে
ইউটিউব লাইভ হচ্ছে, তার পাশে
ওকে
থাকলে সুবিধা হয় বলার সময়
ওকে
অন। একমাত্র যদি না নিজে, মানে নেট অনেক সময় অনেক স্লো হয়ে যায়, তখন অফ করে দিতে হয়। ইফ ইওর নেট ইজ ওকে, নেট স্পিড ইজ ওকে, দেন ইট ইজ
রাইট নাও
বেটার টু,
আমার কোনো সমস্যা নেই,
হ্যাঁ দেন
যদি না
প্লিজ
হঠাৎ
কিপ ইওর ভিডিও অন। ওকে,
ওকে
so I am muting myself. I will formally
ওকে।
start after 5 or 6 minutes when there will be 150 participants at least. Okay. মালবিকা, হ্যালো। একটা রিকোয়েস্ট আছে, সেটা হচ্ছে আমাদের তো ইউটিউব লাইভ হচ্ছে এটার সাথে, যেহেতু সবাই জুম জয়েন করে না, সো ইউটিউবে আমাদের কিছু কনসিস্টেন্ট স্টুডেন্ট রয়েছে। তো যেটা বলছে, প্রণব একটু কথা বলবে তোর সাথে, সুজয় স্কলার, যে ইউটিউবে ওটা একটু বড় হয়ে যাচ্ছে স্ক্রিনটা, সো জুমে তুই যখন স্ক্রিনটা অন করবি, ও বোধহয় ওটাকে একটু ছোট করতে বলছে। আমি মানে ওভার টু প্রণব, প্রণব একটু কাইন্ডলি ম্যামের সাথে ইয়ে করে নে।
তাহলে আমি তো স্টপ শেয়ার করছি, স্টপ শেয়ার করছি। না, সবাই জয়েন করলে দেন।
So let me start formally, because we have already 150 participants, 157 is there, and it will reach approximately to 190 and 192. First, I would like to request all other faculty members of the Department of Zoology, BKGC, whoever is present over here, to on their video, so that I can introduce Malabika with you and you with Malabika, and obviously to dear students also. So this is Dr. Amrita Mukherjee. And where is Moni is also there, Dr. Mohammad Manirujamam. Moni, please kindly, if you can on your video.
Hello, our video is not opening.
Oh, you are.
Madam, video on hocche na, madam.
Okay, I will see. I'll find. Just a minute. Just a minute. I have to do it from another desktop. Just a minute.
It's okay. It's okay now.
Yeah. So now I am going to formally introduce Dr. Malabika Bhattacharjee to all of you, our departmental faculties. Malabika, here is Chinmay Basu, the senior most faculty member of our department. And
Hello, good evening.
Dr. Mukherjee.
Namaskar, namaskar.
And Dr. Manirujamam. And this is Dr. Malabika Bhattacharjee. For students also, I would like to say a few words about Dr. Bhattacharjee. She is the head and assistant professor of the postgraduate department of zoology. Dr. Bhattacharjee completed her graduation from Lady Brabourne College in 1999 and definitely post-graduation from the University of Calcutta. After getting NET, she has completed her PhD from Dr. B.C. Roy Postgraduate Institute of Basic Medical Sciences, Institute of Postgraduate Medical Education and Research for doctoral work. Dr. Bhattacharjee has more than 29 scientific publications in reputed international as well as national journals, which includes International Immunology, Cancer Biology and Therapy, Cancer Investigation, Cancer Letter, Toxicology Letter, Anti-Cancer Research, etc. These are all very reputed journals. She also had an international book chapter to her credit. Dr. Malabika Bhattacharjee is very dynamic lady, I must say. After joining college, she was the principal investigator investigator of two UGC minor research projects. She was also selected as a trainer associate under the UGC scheme capacity building of women managers in higher education, and as a local coordinator, she has organized a 5-day residential sensitivity awareness motivation workshop under the same scheme. She acted as the program coordinator on ornamental fish culture, which is an UGC certified course for a pretty long time. She was also the convener of Institutional Animal Ethics Committee of Thakurpukur Vivekananda College. She has also organized many workshops. She has also received a lot of awards to her own credit. I like to mention a few for introduction. She has got the Best Paper Award in Immunomolecular Profiling of Liver Tumor Induced by a Chemical Carcinogen in the International Conference on Environment and its Impact on Society in 2013. She got the Best Speaker Award in Immune Profiling of Liver Tumor Induced by Environmental Carcinogen in a UGC-sponsored national level seminar on natural and anthropogenic hazards of environments, their impact on biodiversity and conservation, and biotechnological approach for its remedies. She got Best Speaker of the Session in Genetic and Molecular Biology Award section, and also Best Speaker of the Session, and got the medal. Please, really, I know Malabika. She is just a little bit junior from me, and she is very dynamic, student loving, and she is a very, very good teacher. Not only a good teacher, she is also a best researcher, as well as she is, what should I say, she is very much involved with the students with the teaching, learning, and other activities. So I am very happy that Malabika is here, and as we will proceed, we will be able to understand how good teaching skill Malabika really has. So thank you, Malabika, for accepting the invitation. Now it's over to you. You can share your screen and continue. Thank you. So with the kind permission, we are muting all the faculty members of the Department of Zoology. We are here with you, but we are muting ourselves and our video. You please keep your video on. Thank you.
Can I share my video, Papiyadi, now?
Yes, yes.
Can I share my screen?
Yeah.
Okay.
So first you would like to start with the video.
No, I'll start with my presentation. The video
yes,
comes
that
last.
video is okay. You just start sharing your screen. Okay.
But I'm clicking and somehow this thing got hanged.
No problem. You just stop share. First you just stop share. You have your PPT minimized, I think.
Yes,
Stop
yes,
share.
yes.
Stop share. Then again start share. And click on the particular window of the PPT and then share. After clicking on the particular window, the share
It's saying actually that my bandwidth is low. Fine, that is not an issue.
Bandwidth is low. Yeah, it's creating, maybe it's creating problem. Amrita, am I audible?
Yes, ma'am, you are audible.
Okay, you are okay. Then probably Malabika has some problem. Let me find it. Malabika ke ekbar mone hai beriye dhukte hobe. Okay, I am going to make a call to her.
Okay. ল্যাপটপটাই হ্যাং করে গেছে, আমি জয়েন করছি। হ্যাঁ একটা বেরিয়ে ওদের মোবাইলটা ব্যাকআপে রেখেছে, আমি চেষ্টা করছি কম্পিউটার থেকে না হলে, আমি তখন ওর মোবাইল থেকেই করছি। তো হ্যাঁ না আমি কম্পিউটারটা কানেক্টিং হচ্ছে, দেখি আর একবার কম্পিউটার থেকে করলে বেটার হয়, কার্সারটা নয়। হ্যাঁ হ্যাঁ কার্সারটা নড়ে না, ঠিক আছে আমি দেখছি করছি আবার আবার।
ওর মোবাইলে অন আছে কম্পিউটারটা। Participant, actually Malabika ma'am is on with the mobile. Her computer is disturbing a little. Within 2 minutes, she is joining either by the computer or by the mobile. She will share the screen. She is trying to share the screen with the computer. Otherwise, she will share the screen through the mobile. She is on in mobile, but computer has hanged somehow. Just a minute. এসএস এম আর এমএম একটুখানি সবাই খেয়াল রাখো, অ্যাটেন্ডিজে মালবিকা আর একবার জয়েন করলে আমাকে একটু বলবে। আমিও দেখছি মিস
আমি আমি জয়েন করে গেছি। তুমি
কোনটাতে, কোনটা দিয়ে?
ল্যাপটপ দিয়ে জয়েন করেছি।
দাঁড়া দাঁড়া, ও হ্যাঁ হ্যাঁ ঠিক আছে ঠিক আছে, দাঁড়া দাঁড়া এক সেকেন্ড।
শিওর।
ইয়েস হ্যাঁ দেখ তো এবার ঠিক হচ্ছে কিনা। তো ইয়েটা অন কর, ভিডিও ভিডিও ইয়া ইয়া এস ইয়েস ইয়েস ইয়েস। এই ল্যাপটপে খুব সমস্যা, হ্যাং হয়ে যাওয়া। Yes, now you share. Now you share.
আচ্ছা now I can share. Okay, share screen. Yes, it is coming. একবার স্টার্ট হয়ে গেলে বাস।
হ্যাঁ হ্যাঁ একবার
Okay, the screen is
that's
visible.
fine. Yes, absolutely, absolutely.
And I'm starting.
Yeah. Okay, it's
Good
okay.
evening. At the outset, there's echo. Echo হচ্ছে না?
Yes, because you have two device on. Keep one device to some other room.
Yeah, but I have that on Bluetooth.
Somewhere else at least.
Now the sound is fine?
Fine, fine.
Okay. First of all, thank you.
No, no, echo is going on.
Somebody's actually kept his or her.
No, I am. Let me mute.
Now I think so it's fine.
No. Participants are actually all muted because it is in Zoom, and we are also, except me, everybody is muted. It's better that you off your phone. If there will be some problem, you can rejoin with the phone. এটা আমি দেখেছি আমার আমার ঘরে, আমার যেহেতু এই ডেস্কটপটা যেটা এখন কথা বলছি ওটাতে ওয়েবক্যাম নেই, পাশে যদি মোবাইল অন রাখি কন্টিনিউয়াস ইকো হতে থাকে, মানে অনেকটা দূরে নিয়ে গেলেও হয়। কেন
আমি দিয়ে এসেছি কিন্তু
স্টিল ইকো হচ্ছে, কিন্তু তুই মোবাইলটা একবার পুরো বের করে বেরিয়ে যা, অসুবিধা নেই তো, আমরা পরে ঢুকে যাব প্রবলেম হলে। লেট মি সি। অ্যাটেন্ডেন্স সবাই অফ আছে, অ্যাটেন্ডেন্সে সবাই অফ আছে।
হ্যাঁ এবার হচ্ছে
না না, ওটা হয়, ওটা পুরো বাড়ির মধ্যেই ওই প্রবলেম।
Gone out of that.
হ্যাঁ হ্যাঁ, ইটস ওকে ফাইন।
Okay, anyway. So with a lot of technical glitches that happened, anyway, I'm sorry for all the technical glitches. But at the outset, I'd like to thank, it was a very generous description given by Papedi about me. So good evening to all. And at the outset, I'll thank the principal, Dr. Bhattacharjee of Vijay Krishna Girls College, Howrah, for inviting me as a resource person to this 2 weeks international student development program on advanced learning and career guidance. I'd also specially convey my heartfelt thanks to Dr. Papia Ghosh, or rather Papia D to me, for inviting me to this digital platform. And with this, I start my today's lecture, or rather a talk, an interactive talk, the CRISPR-Cas system structure and mechanisms. Now, CRISPR-Cas is a defense system in which bacteria use RNA molecules and CRISPR-associated Cas proteins to target and destroy the DNA of invading viruses. It was in fact in 2012, in a landmark paper, 3 scientists, we'll come to that, I'll show you the pictures later on, Doudna, Carpentier and Jinek showed that they could use this system to cut the genome at any place they wanted. And it was this paper actually which gave this CRISPR-Cas system a huge impetus. It is now one of the hottest field. In 2011, there were only as few as 100 published papers. But after the seminal paper of Doudna and Carpentier, where they said that this system can be used for genome editing, in 2018, the number of published papers in this sector became 17,000. So with this brief introduction to the topic, and I have shown a picture of a baby here. So this system is theoretically, it can edit the genome of any organism from wheat to mosquitoes and even humans. Now, before getting into the genome editing part or the proper part of CRISPR-Cas, I just wanted to share with you all that what was the road to this particular method of genetic engineering. So if I think of biology and how biology has made leaps and bounds in various biotechnological fields, all started with the very famous rules of biology. In 1851, we all know about this, Darwin proposed origin of species. And then 1856 to 66, in the field of genetics, it had a huge impact, the Mendelian inheritance. In 1868, Frederick Miescher first discovered nucleic acids. He initially called it nuclein, but later on it came to be known as, that discovery is now what we know as DNA. So these were some seminal discoveries that laid the foundation rules of what we today now see as genetic engineering. Coming to the information part, what were the informations that were needed to finally reach to this genome editing section? The informations that were needed, that were discovered and that helped us was in 1944, Avery, MacLeod and McCarthy, many of you have studied this famous experiment, first time showed that DNA is indeed the genetic material. In 1953, Watson, Crick and Franklin discovered the DNA structure, and with this, it started the age of molecular biology. In 1961 to 67, the genetic code was discovered. Now, there were certain basics, certain tools also that came up, which ultimately led us to this field of genome editing. What were the tools? The first tool that helped, that gave a huge boost to genetics and genome editing sector was the discovery of the restriction enzymes. In 1977, Sanger sequencing was discovered. In 1983 came this very famous and most used discovery, the PCR. In fact, it became so famous during that time that it was colloquially called a people's choice reaction among the molecular biologists. And finally, in 2003, the Genome Project developed. And now we come to this era of genome editing. I'll not be talking about the zinc fingers or the talons today. I'll totally focus on CRISPR because today I'll be dealing with CRISPR-Cas system. Now, just to give a brief intro to the genome editing, the 3 basic genome editing tools. These 2 genome editing tools are basically protein based, but the one we are going to realize, we're going to walk through slowly and discover the various parts of it and the basic strategy in which it works. CRISPR is basically RNA based. Now, a very brief history. I'll just walk you through the very brief history of CRISPR. The discovery of CRISPR was, in fact, it was accidentally discovered in 1987 in the E. coli model where Nakata et al. He was investigating on alkaline phosphatase and the IAP gene in E. coli. And there he saw that there were repeats, and these repeats were spaced apart by non-repetitive sequences. It was from 1987 to 2002. A number of names were given to these repeats, but the word CRISPR, they were officially termed CRISPR in the year 2002, and it was in the same year that the classification system of CRISPR was done based on the Cas genes. So that was also discovered in that same year. In 2005, this is a very famous work of Mojika. I'll be showing you his picture next slide. He discovered the CRISPR array that originated from bacteriophage, and he was the one to propose that this CRISPR locus is somewhat related to the adaptive immunity of bacteria. In 2007, it was for the first time experimentally proved in Streptococcus thermophilus that it indeed contributes to bacterial adaptive immunity. In 2008, the CRISPR RNA were derived from the spacers. We'll be coming to those definitions slowly. Then another important discovery in this segment was done. That was the tracer RNA and the Cas9 target discovery was done. 2013, first editing in mammalian genomes. 2015, CRISPR used in human embryo, but that was an unsuccessful attempt. And thereafter, the CRISPR techniques have been modified. In 2016, National Institute of Health actually first time approved human trials with CRISPR. And interestingly, they said that these CRISPR edited crops would not be considered as genetically modified organisms. And finally, in 2017, a variety of ongoing trials were approved by this CRISPR-Cas mechanism. So this is Mojika et al., who for the first time in 2005 proposed the importance of CRISPR in the immune function of bacteria. So it plays a role in adaptive immunity. And it was in 2002, it was named as clustered, regularly interspaced palindromic repeats. I'll be breaking those words and explaining. And these are the three famous scientists who in 2012, followed by 2013, in these two papers, provided evidence that genome editing could be very easily done by the CRISPR-Cas9 system. So this is Jennifer Doudna, Zhang, and Emmanuel Charpentier. And interestingly, since you have studied the variety of DNA repair pathways, just to point it out here that this form of genetic editing uses two basic DNA repair pathways. One is the non-homologous end joining pathway where the DNA is cleaved and then the blunt ends are ligated, but this is an error-prone repair pathway because insertion or deletional mutations are often introduced. That is together called as indel. And the other is the homology-directed repair pathway where there is a repair template and the repair is done. So if we want in the cleaved end, depending on the repair template, a defined sequence can be inserted. So this is the two repair pathways this CRISPR-Cas system uses for editing purpose. So this is the bacteriophage, and it attacks the bacteria, and the CRISPR-Cas is basically a basic protection of the bacteria to the virus. It's a basic immune protection. This is a very simplified scheme. So those bacteria without the CRISPR-Cas system, when the bacteriophage infects the bacteria, the virus infects the bacteria, inserts the viral genome. So those without the CRISPR locus, they become susceptible to the bacterial genome mediated cleaving or final lysis of the bacteria. But as you can see, some bacteria do have a mechanism of protection. That is, as I said, we'll be discussing the CRISPR-Cas system. And via this system, what happens whenever an invading bacteriophage, a virus, inserts its genome, then what does this molecular scissor do? It just chops off the viral genome, resulting in double-stranded breaks in the viral genome, thereby protecting the bacteria. So this system somehow, this CRISPR system is somehow, first see, it recognizes certain sequences, cleaving the sequences, thereby protecting the bacteria. Now coming to the most simplified picture of the CRISPR-Cas system, I'll be going through next 3-4 slides where I'll be increasing the level of complexity of the pictures. This is the most simplest of the pictures, and I just want to give you a glimpse of the parts of the CRISPR-Cas system. To be very generalized, the CRISPR-Cas system has 3 functional steps: acquisition, expression, interference. Now look, this is a bacterial cell. And let us imagine that a bacteriophage has invaded this cell. So the moment a bacteriophage or the virus of the bacteria infects the cell, it introduces its viral genome within the bacterial cell. Within the bacterial cell, if the bacterial cell has a CRISPR locus, so what happens? The moment the viral genome is inserted, the CRISPR locus comes into action. Now let us see. This is a CRISPR locus. So it's nothing but a stretch of DNA. And as you can see by the name, clustered, regularly interspaced, short, palindromic repeats. So this is a repeat sequence. This is a repeat sequence. The repeats are palindromes. Now what does the word palindrome mean? It is such a sequence which when you read from 5' to 3', or from 3' to 5' reads the same. A beautiful palindromic word is madam. So if you read it from left to right, it is M-A-D-A-M. From right to left, it is again M-A-D-A-M. So these are palindromic repeats. Now, if you look into the arrangement of the DNA, there is a leader sequence. I'll be getting into the details of the function of the individual components in my next 2 or 3 slides. For simplification, this is the black one is the leader sequence and this is the DNA. Within the DNA, you can see the red is the repeat part and the green is the spacer part. Now, what is this spacer part? Repeat part, you can just understand that these are simple repeats which are placed in a regular manner with some intervening spacers. Now, what is the spacer part? Spacer part is nothing but a small amount of the viral genome that has been cleaved and inserted into the bacterial DNA. So this spacer part is nothing but a part of the viral DNA that has been cleaved and inserted. For an RNA genome, a cDNA copy is made and cleaved and inserted. So the spacer is nothing but a cleaved segment of the viral genome that has been introduced into the bacterial DNA. How it is done, I'll be coming up later. So we are having repeats and spacers. Repeats are unique to the bacteria. Spacers are obtained from the viruses. So this part is known as acquisition. So the bacteria is taking fragments of DNA from the foreign genetic elements that have entered the cell. Coming to the next part. This RNA is now being transcribed. So this is the biogenesis of the CRISPR RNA. So that is the expression part. Not only is the expression part involves not only the biogenesis of the CRISPR RNA, but it also fragments the CRISPR RNA. So together, this is the expression part. Now, if we imagine that this is a spacer taken from this virus. So this is a fragment of DNA taken from this virus. And when we have matured small CRISPR RNA fragments, these fragments actually contains a piece of a DNA which has actually been taken up from this virus. It has come to this RNA via this DNA intermediate. So it has this fragment of DNA taken from virus entered the CRISPR locus as spacer, was transcribed as the CRISPR RNA and is then matured in form of CRISPR RNA. Now comes the interference part. CRISPR RNAs do not act alone. They have a partner. And now we come to the next part, the Cas, CRISPR associated protein. So the Cas system. So as I said, they do not act alone. They have partners which are protein partners. The RNA and the protein partners bind. And if this same virus then infects the bacterial cell, this sequence will be able to identify the DNA. Actually, it has originated from a segment of DNA from here. So it will be able to identify the target sequence DNA in the viral genome. It will go and bind to the viral genome, bringing about the cleavage of the viral DNA. So when the virus enters the first time, the spacer is acquired and serves as a memory, so that if the same virus infects second time, the memory RNAs now go and bind with the target DNA sequence and cleave the virus. Coming to this picture. So we now move to a bit complex picture. If we look into this picture, as I said, it has adaptation 3 parts. As we discussed earlier, there is an adaptation part, there is an expression part, and there is an interference part. So let's go step by step. First part is the adaptation part. In the adaptation part, let us look at the CRISPR locus. Now, as I said, the CRISPR-Cas system are widespread and are found in 90% of archaeal and 40% of bacterial genomes. In the current classification system of CRISPR, it is totally based on the number of Cas genes that are available. There are basically 2 classes of CRISPR, class 1 and class 2. And they are further divided into at least 5 different subtypes. Now, talking about the repeats. Now, if we look into this CRISPR locus, as you can see, the CRISPR locus, there are gray blocks and beautifully colored blocks. Let's concentrate on the gray blocks. The gray blocks are actually the CRISPR repeats. Now, the CRISPR repeats in sequence genomes, in genomes where the sequence have already been done, they range from the size of 23 to 25 base pairs. CRISPR repeats were classified into 12 major and 21 minor groups on the basis of sequence similarity. The repeat sequences, the gray blocks, again, the gray blocks, the repeat sequences are highly conserved. Repeat sequences are highly conserved within a given array. And although certain deviations have been found, the repeat at the distal end. So if I think of the CRISPR locus, this is the leader. So the nearest repeat array is called the proximal end, and the farthest repeat array is called the distal end. So it is often observed that the repeat sequences at the distal end has become degenerate. What does the word degenerate here means? They have become structurally to some extent different, but functional wise, they still perform the same function. So these are the repeats, the gray blocks. Again, I'm repeating, the gray blocks are the repeats. And in between the gray blocks, we are having some very beautiful colored blocks, pink, orange, lemon yellow, yellow, green. So these are the spacers. The spacers are located within a repeat array. They are more or less similar in size with a few nucleotides and carry unique sequences. Obviously, they will carry unique sequences because these spacers are nothing but fragments of DNA that has been acquired from various viruses that had attacked the bacteria one after another. We have to keep one thing in mind, that if in this CRISPR locus, this is the proximal end, and if this is the distal end, the purple color, as you can see, the purple color, so that spacer is the oldest one to be acquired. And if you look to this pink color, this is the newest one to be acquired. So so as we move down the leader from the proximal to the distal end, the newest ones are added to the leader proximal end, and the oldest ones are found on the distal end. So for this particular CRISPR array, I can say that the purple one came first, then the blue, then the different shades of blue, then the 2 shades of green, the 2 shades of yellow, orange, red, and pink. So this is the story about the spacers. What is the exact function of the spacers? The spacers provide resistance to any foreign nucleic acid that contains the same or very closely related sequences. Genes upstream of the repeat This structure has not been shown, so I'm not going into that. I'm just saying that there are genes upstream of the repeat and the spacer sequences which code for the CRISPR associated proteins. We'll be taking that up in the next 2 slides. So we have understood what is a CRISPR locus basically consists of. It is the repeats and the spacers. And just in my previous slide, as I had said, that this CRISPR locus is transcribed to give us a CRISPR transcript. And the CRISPR transcript is then producing mature CRISPR fragments, which then complexes with the Cas proteins to form an effector complex. And as I said, if the same virus enters here, then this effector complex, this is basically a CRISPR ribonucleoprotein complex. There is an RNA part to it and there is a protein part to it. So this CRISPR ribonucleoprotein complexes will then bind with the sequence from which it had originally it had been acquired and will bring about the cleavage of the foreign nucleic acid. There are 2 things which I'm not mentioning here, even though it is here in the slide. I'll come to that later on. This is the PAM sequence. I'm not describing it out here in this slide. And there's another thing called the protospacer. What is a protospacer? The protospacer is actually the candidate pre-spacer which was acquired. And in the later stages, when this virus will attack again, the spacer will target this protospacer sequence. So that is the protospacer. Now let us come to this part where it has been shown beautifully that how a candidate pre-spacer is acquired. See, there are certain enzymes that belongs to the Cas family. They cleave the incoming genome, as you can see. So this is the candidate pre-spacer. And this pre-spacer is then incorporated into the bacterial CRISPR locus. But we have to keep one thing in mind. Whenever a pre-spacer is introduced into the CRISPR locus, target site duplication is occurring. That is, it is also duplicating a repeat area. So see, this was the original repeat. The pre-spacer got inserted and a repeat of the CRISPR repeat has also been done. So how does this happen? First, let's deal with that how does this happen? This is a beautiful paper. This is specifically, this was worked on the E. coli CRISPR structure, where the CRISPR array was 28 nucleotide long. So in this structure, first let us see what the structure tells us. This blue color is a 80 rich leader sequence. And this is the CRISPR locus. The green loops are the CRISPR repeats. They had crystallized and deduced the structure of this, and these green structures, this was actually the crystallized structure of the CRISPR repeat. So these green loops are the CRISPR repeats. And within these green loops, we are finding that spacers are integrated, as I said. Now what happens? Whenever there is a new spacer acquisition, you see, there is a 3' OH, space where there is a OH, yeah, there's a 3' OH at 2 ends of the DNA. So as you can see, I have given a very comparative analysis of how this is being done by showing you the cut and paste mechanism of transposition, with which many of you are very familiar. In the cut and paste transposition system, what is seen? Whenever a transposon is inserted into a target DNA, you see the O of the 3' end actually attacks the target site, cleaves the target site. So the transposon gets inserted and there is a duplication of the target site. So it's a very similar mechanism to it. The 3' OH is actually inducing a cut in the CRISPR repeat, getting itself inserted, resulting in a target site duplication. So that is how, when a spacer is integrated into the system, the target site duplication is also occurring. But this area has not been worked out in very much detail. If you look into this, this is again the same mechanism. How this talks about two mechanism, and this particular picture I have taken from a paper of Dudna herself. Now, as you can see, how is the protospacer selected? If you remember, the sequence that is to be acquired from the viral genome is actually acting as the protospacer. So, as I said, that basically there are 5 types of CRISPR mechanisms. So they have shown in this particular paper, they showed 2 basic type of system. In type 1 system, which is known as primed adaptation, the CRISPR-Cas enzyme system, I'm not going into the name of this big enzyme system is known as cascade. So it's CRISPR associated complex of antiviral defense. It contains a number of subunits, and together it is known as cascade because it becomes very big in size. And the RNA, CRISPR RNA, they bind to the viral genome. As you can see, binds to a mismatched target. So they bind to the target. The nuclease and the helicase, Cas3 is another enzyme, as another protein associated with the CRISPR type 1 system. So as you can see, the Cas3 is recruited to the target site, and then the Cas3 moves along the viral genome, and as you can see this, and a new location is selected. In this location, these 2 Cas proteins are brought into it, and this Cas1 and Cas2 protein cleave out this. So this is basically the model concept of how it might go. The exact mechanism of how it is done is yet under work. This is the type 1 naive system. This happens in E. coli, where the E. coli involves the nuclease helicase REC BCD pathway. The degradation products appear to serve as substrates for Cas1 and Cas2. However, these degraded products are of variable length, but how do the REC BCD pathway convert them into double-stranded prospacers of similar size has not been worked out. This is for the type 2 system. You have heard about this protein Cas9 for some time. In this case, as you can see, the Cas9, the type 2 system, the CRISPR RNA enzyme has bonded here. The complex with the whole complex with this protospacer bond recognizes this target site by a presence of a green color block you are seeing, which is named as PAM. And once the target site has been selected, then the 3' OH of each protospacer strands makes a nucleophilic attack on the repeat backbone. So it's something like the one I showed you just, that the 3' OH actually makes the attack. So it's something like this that happens. And in the type 2 system also, the PAM recruits the Cas1 and the Cas2 and cleaves out the protospacer. Now, this is the picture very similar to just now what I showed. So the Cas1 and Cas2 have acquired the protospacer. Now they need to insert it. So just now I showed you the picture. So they bind with the repeat region, target the repeat region, induce a single-stranded break, and they insert the repeat, bringing about target site duplication. As you can see, is the 3' OH that brings about a single-stranded staggered cut, introducing the spacer, bringing about duplication of the repeat segments. So this is how the protospacer selection has been assumed. It has been assumed that this is how the protospacer selection is done. And this is the most probable mechanism of insertion of the protospacer in between the protospacer into the target site, into the CRISPR locus, resulting in a generation of target site duplication. So the target site duplication is nothing but the duplication of the repeat site. Now let me come more broadly to this structure. So as I said, this is the CRISPR locus. This is the leader sequence which we have heard of, and this is the Cas genes. Till now, I had told you about the Cas genes, but I had not showed you how they reside. So they reside upstream of the leader sequence. We have heard what are the diamond blocks are the CRISPR repeats, and the colorful rectangular blocks are the spacer sequences that has been derived from the protospacer. Now let's get into some Another part of the CRISPR story is the Cas genes and the leader. Now, the leader is very important. The size of the leader ranges from 100 to 500 base pairs. Leaders are oriented specifically with respect to the repeat sequence of the adjoining CRISPR locus. So, they are oriented with respect to the adjoining CRISPR locus. Leaders are non-protein coding and often carry low complexity sequence regions. The leaders has basically two functions. The first function of the leader is to provide the promoter element for transcription of the CRISPR locus. The second function of the leader is that it sits at a site, it adjoins the site where new spacers are to be added. I had said you in my first or second slide that there is a definite orientation to the adding of the spacers. So the newest spacers are added to the leader proximal end, while the oldest spacers are found in the leader distal region. Now coming to the CRISPR RNAs. As you see, the CRISPR locus is transcribed to give the CRISPR RNA, which is one long molecule that is cleaved in middle of each repetitive sequence. So keep this in mind, it is cleaved in the middle of each repetitive sequence by Cas proteins with ribonuclease activity. So many of the Cas proteins have ribonuclease activity. As a result of this cleavage, this converts the pre-CRISPR RNA into individual specific segments known as CRISPR targeting RNA or cRNA. So the cRNAs are the mature forms, are the CRISPR targeting RNAs. The size of the mature CRISPR RNA ranges from around 30 nucleotides to more than 60 nucleotides. Another thing that is to be mentioned here while talking about the RNA itself, they contain complete or partial spacer sequences and portion of flanking repeats. So this processed RNAs have a part of the flanking regions of the CRISPR repeats and usually the partial or the complete part of the spacer sequences. During interference, what is interference? That is interfering with the growth, interfering when a new DNA has been introduced from the same virus which had infected it earlier. So when these RNAs go and bind to this new DNA, ultimately cleaving it and thereby preventing viral growth, so that is interference. So we have to keep that in mind that during interference, the spacer sequence pairs with the complementary protospacer sequence, while the residual repeat sequence, as I said, there are CRISPR repeat sequence that are flanking the spacer sequence. So the residual repeat sequence probably provide attachment site for the Cas proteins to help in this interference procedure. So this is the role of the CRISPR RNA. Now coming to the Cas proteins. As you can see, there are three kinds of genes involved in the Cas system, and they are in different shades of blue. The A1, the genes belonging to this shade, they are involved in acquisition, and the most known Cas protein that is involved in acquisition or acquiring the protospacer is Cas1 and Cas2, just as we saw it in the previous slide. Next comes the I genes. The I genes produce those Cas proteins that help here in the interference part, okay? And these are the P genes, Cas P genes, they help in expression. So these Cas proteins actually help in the processing of the pre-CRISPR RNA to the mature CRISPR form. So there are three categories of Cas genes. And the Cas protein, as you can see, has acquired the protospacer. So that is adaptation, has inserted the protospacer. You can see the P genes. Once the CRISPR RNA has been transcribed, the P genes producing the Cas proteins, they are helping in processing. They are helping in processing of this CRISPR RNA into mature CRISPR RNA. And finally, if you see the Cas genes belonging to this I category, they are helping in interference of the viral DNA that plans to invade the bacteria and wants to make a colony of its own. So I have discussed right now most of the components of the CRISPR system, but there is one very, very vital component which I have not introduced till now. This component is very, very vital in the sense because CRISPR was initially discovered as when it was in 2005, when Mojika finally said that CRISPR has a role in immunity. So it is protecting the bacteria from being broken down by a bacteriophage. Now the question comes that since these RNAs, the CRISPR mature RNAs, are actually made from these DNAs. So when we are having this matured CRISPR ribonucleoprotein complexes, there might be a tendency of these matured complexes to come and bind to this DNA itself. So instead of identifying the foreign target sequence, since it has been born from this sequence, it will come and identify the bacterial DNA itself and will cleave the DNA. So you can see how brilliant a mechanism a small single cell microbe has applied to protect its spacer sequences from this cleavage. And what is that? Now we come to this red molecule. As you can see, this is known as the protospacer adjacent motif. So whenever the Cas1 and the Cas2 proteins identify a protospacer, they do not just identify any DNA segment. They have to identify a DNA segment that has a PAM sequence flanking it. So the PAM sequence, it's a sequence, okay, is flanking this protospacer. Now, PAM are very short sequences, 2 to 6 bases that are present in only foreign nucleic acid. There is no PAM present in the bacterial DNA. So they are present only in the foreign nucleic acid. For example, in case of Cas9, you have heard about this particular molecule quite enough. That is useful for genetic engineering, recognizes a 5' NGG3' sequence where N can be any nucleotide. Some effector complexes require both the binding of the CRISPR RNA to the protospacer and the recognition of a PAM by other components of the effector system. So whenever this CRISPR RNA thinks of if there is a foreign DNA that has been inserted and the CRISPR RNA is now mobilized, the effector complex is now mobilized, the CRISPR RNA recognizes the protospacer region, while the effector complex, that is the protein part and to some extent the RNA part also recognizes the PAM region. So the presence of PAM not only helps in locating the target nucleic acid, but since this PAM are not present into the bacterial DNA, it also protects the bacterial DNA from autoimmune cleavage. So if this sequences, since these red color sequences are not present here, so this mature form of RNA will not recognize them. It will recognize only those DNA having a PAM followed by a target sequence. In this way, autoimmunity is being prevented in a bacterial system. The question is that, do all systems have PAM? No. All CRISPR systems do not have PAM. For some, it has been seen that in these cases, a few nucleotide mismatches within the flanking regions must be present between the CRISPR RNA spacer and the target sequence for CRISPR mediated degradation to occur. So the CRISPR RNA, so what does it actually say? The CRISPR RNA goes and binds with the protospacer. And if there is no PAM, then there must be some mismatches occurring. I'll show you in a later figure. So there is actually a 5' handle or a 3' handle. I'll come to that later on. So there is not a perfect pairing. At the 5' end or the 3' end, there should be a mismatch. And if this mismatch is found, the CRISPR RNA will realize that this is a foreign DNA. But while bonding, if there is no mismatch to be found, that means somehow mistakenly the CRISPR RNA has bonded with its own DNA, mother DNA, to which it is completely complementary, and as a result of which the DNA or the RNA is considered self and cleavage is not occurring. So there's another strategy. The one is the PAM strategy. The second is that there are certain mismatch of sequences. If there is mismatch of sequences, the CRISPR RNA will realize that this is a foreign DNA. If there is no mismatch, then the CRISPR RNA will assume that this is the self DNA. This is the self DNA, so have bonded with the self DNA, so it will not cleave. And finally, for class 2, as I said, there are two classes of CRISPR, class 1 and class 2. For class 2 type 4 system, even they do not have PAM. They use a sequence which is known as protospacer flanking sequence, PFS. The type 6 systems only cleave RNA. So to differentiate between PAM, which is found in DNA, to differentiate it from PAM, it was given a new name, though it performs the same function as that of PAM, and this name was protospacer flanking sequences. So this is basically in details about the different components of the CRISPR-Cas system. So there is this foreign DNA, we talked about PAM, we talked about the protospacer, which is the target sequence, which is acquired by the Cas proteins, which is inserted into the CRISPR locus by a mechanism I just now said, resulting in target duplication of the CRISPR repeats. This is the leader sequence, and the promoter is present within the leader sequence, and this is the proximal end of the leader, this is the distal end of the leader. This after the acquisition is done, this is the CRISPR RNA that is produced, that is the primary transcript, which is then processed, and finally if the same virus infects the cell, then the spacer sequence will recognize the protospacer sequence in context with the PAM, and then only will cleave the invading DNA. So this is the generalized process of how the CRISPR-Cas system occurs within a bacteria. Now let's get into some details. So as I was going on saying that there are basically two classes of CRISPR, and there are 6 types of CRISPR. The most well-known being the class type 2 system, which uses Cas9 and the CRISPR tracer RNA system. This being the most famous because this is the CRISPR-Cas9 is the system that is being widely used for genome editing strategies. We have the type 1 system is also quite famous, and there is this type 3 system, which just to note one thing, both the type 1 system and the type 2 system, which has gained momentum, both cleave DNA. So the target is DNA, but in case of type 3 system, the target is both DNA and RNA. And as I said, there's a generalized mechanism. So what does the generalized mechanism say? A spacer is obtained from the genome. The spacer is inserted into the CRISPR locus. The CRISPR RNA is synthesized. The CRISPR RNA is processed, and the CRISPR RNA and the Cas protein form complexes. And now they go and attack the target DNA if that has been inserted in context with a identification sequence, which can be a PAM, which can be a mismatch, which can be a PFS. So based on this, so these are the various, so that is a general rule. Now the modifications, where are the modifications being involved? The modifications are being involved in the number of Cas proteins that are being involved and the category of Cas proteins that are involved. That is where the modification is occurring. But the general protocol is that. So it's something like that, that we have for since there are a lot of UG students, when you read a syllabus, there's a general protocol. So you have to give 50 marks for molecular biology, you have to give 50 marks answer for immunology, you have to give 50 marks answer for developmental biology. But so that's the general protocol, that you have to sit in an exam. That exam follows a 2-hour definite pattern. You have to take theory papers and you have to see the question paper and write the answer. Where is the difference? The difference is in the subject matter, something like that. So there's a general protocol, and based on the number of proteins that are involved, we are having these different types. This particular picture is also from Dudna, and this paper, if people are interested, they can go and read this paper. This paper is available. This is available. This is open access paper, and this is available. Now, if you look into the Cas genes, so the CRISPR systems are extremely diverse and can be classified into 6 major types based on the number of Cas genes present. Now, there are certain uniqueness to the system. For example, tracer RNA is found only in type 2 system. Till now, I have not talked about tracer RNA. So I will be talking about tracer RNA when I do that type 2 system, because I'll be doing that in a bit detail, as that is the system that is used for genome editing. And there is another Cas that is also unique in nature, the Cas6, that is only found in type 1 and type 3 systems. So these are the variety of Cas genes that are found, as I showed you in the picture, upstream of the leader element. And they have a variety of functions. The colors do mean something. The ones that are red in color, for whatever system, the ones that are red in color are involved in interference. The ones that are yellow colored, they are involved in biogenesis, that is maturation process. And those that are blue, I wouldn't say this color, those are involved in adaptation, that is acquiring target sequence from the virus, which is now called the protospacer, and incorporating it within the bacterial DNA. So, and this is the general strategy. As I said, the general rule is the same. Now, there are variety in the number of genes and in the different types of proteins that are produced. So, as I said, these are the 3 famous basic subtypes, and I have showed you the same. The process is basically the same. We have a CRISPR RNA and a group of proteins, and they recognize the PAM and target the nucleic acid type 1. Type 2, it's a single protein and the CRISPR RNA and the tracer RNA present here, and then the mature form target the genome by recognizing the PAM. And similarly, here in type 3 also a number of enzymes are involved and they target the genome, but here the genome is targeted, as I said, by understanding that whether there is a sequence mismatch in the flanking regions or not. And these are the variety of enzymes involved, the CRISPR proteins, and most of them have nuclease activity, ribonuclease activity. Another important enzyme that has not been mentioned earlier is RNase 3. This is important in the sense that when we will be doing the type 2 system, we will see that this enzyme plays an important role in processing part of the RNA for type 2 system. So it processes the tracer RNA and assessing CRISPR maturation. So this is simple RNase 3 of present in the bacteria. And now coming to a bit detailed structure. So as you can see, this is the type class 1 type 1 CRISPR ribonucleoprotein effector complex. This is the target DNA. See, this is the PAM, as you can see. So in case of type 1 includes the CRISPR RNA, and this total complex is known as the CRISPR associated complex for antiviral defense, cascade. It does not involve one protein. It involves quite a large number of proteins. It is a multi-protein effector complexes. So this complexes recognizes the DNA strand through the complementary base pair of the CRISPR RNA, and then the Cas3 induces the nucleotide break, as you can see here. Now, this, as you can see, this is another system. This is class 1 type 3 system, where a coupling phenomena is observed, where in case of viruses which have already started transcribing, so there is a growing RNA and there is the viral DNA. This type 3 coupled system can target both the single-stranded RNA and the single-stranded DNA. So there is the breakage in the DNA and also breakage in the RNA. So it's cleaved, the DNA is also cleaved, and the RNA that is being actively transcribed by the viral DNA is also cleaved. So as I said, this is another picture of the type 1 cascade system, and as you can see, there are it's a big group. You can see the number of proteins involved. This is the invading nucleic acid. On recognition by the PAM system, this whole complex goes and binds here, and finally the Cas3 mediates the cleavage. Now this is the class 2 type 2 system. In the class 2 type 2 system, as I said, I'll be doing this in a bit detail because this is the main system that is used for genome editing. What happens? There, let us look at the system first. There is a CRISPR RNA, which we have studied by now. There is a target sequence or the protospacer of the invading genome, and there is a PAM sequence for identifying that invading sequence. Okay. Now let's look at this picture. So a bacteriophage has infected and has inserted its DNA. The DNA is fragmented, and the fragmented DNA is acquired by the Cas1 and Cas2 complex. So it is being acquired by the class 1 and class 2 complex. Now what happens? Once the spacer acquisition has done, if you remember, there will be spacer integration. So the spacer will be integrated in this repeat area. Okay. So the moment the spacer is to be integrated into this repeat area, what happens? See, the spacer is integrated with the duplication of the repeat area. You can see the spacer has been, this is the repeat area, and the spacer has been integrated with the duplication in the spacer area, in the repeat area. Now this brown color is the leader sequence. Upstream of the brown color, we are having some blue segments. These are components of the Cas gene. And upstream of that, we are having now, till now we have not heard about this word, that is the tracer RNA. Now, what happens? So, once the integration of the spacer has been done in between the two repeats, as you can see, the pre-CRISPR RNA is synthesized. Simultaneously with the pre-CRISPR RNA, the tracer RNA is also being synthesized. This arrow actually shows the direction in which the promoter will move, resulting in the synthesis of the RNA. So, along with the CRISPR RNA, the tracer RNA is synthesized, and now you can see we have the bacterial RNase 3 and the tracer RNA. The bacterial RNase 3 and the tracer RNA will now, with the help of Cas9, will now cleave this fully transcribed CRISPR locus into, that is a fully transcribed CRISPR mRNA, into the mature forms. So we have the spacers and we have parts of the CRISPR repeats and the tracer RNA. So this is our effector complex. So if the same virus now attacks the bacteriophage, since we had a spacer in any one of this spacer will now recognize the protospacer in context with PAM and bring about the cleavage of the invading genome. So there is another picture. This is a enlarged picture. So I showed this one picture, as you can see. So this is the bacteriophage. This is the protospacer PAM, integration of the protospacer by recognizing integration of the protospacer. And then, as you can see, the tracer RNA is synthesized. The tracer RNA, and I said RNase 3, along with caspase, helps in forming mature CRISPR RNA, and the mature CRISPR RNA will now bring about the interference. This is a very schematic representation of the domain of the representative Cas9s. So these are from 4 bacteria, and these are the representative of the domains of the Cas9. And now coming to the importance of Cas9. We have studied the systems, we have not in detail studied, but somehow you people have got an idea about the 3 basic categories, type 1, type 2, and type 3. And till now what you saw, if we think about the number of Cas proteins involved in type 1 or type 3 as compared to type 2, you will see that there are more than 1 Cas protein involved in type 1 and type 3. But in case of type 2, there is only 1 Cas protein, and that is Cas9. So that's the beauty of the system, and that's the most simplest of the system. So there is only 1 protein. Now somehow, and what did we see in the earlier picture? Just in the earlier picture, we saw that, see, if you see this picture, so there is 1 blue protein, that is Cas9, and we have a CRISPR RNA and a tracer RNA. Now, in case of the CRISPR RNA and the tracer RNA, what happens during interference? During interference, the tracer RNA is expressed and then processed by the ribonuclease RNase 3. Now, a complex of this tracer RNA, the CRISPR RNA, and the Cas actually cleaves the invading genome. So the tracer RNA and the CRISPR RNA do play a role along with Cas9 in recognizing this PAM. So the importance of tracer RNA lies in the fact that it helps in recognizing the target sequence. So when we think of the Cas9 system, so we have 2 RNA moieties, the tracer RNA, the CRISPR RNA, and there's 1 protein. If somehow we can introduce an artificial linker in between the CRISPR RNA and the tracer RNA, and then convert it into a single guide RNA. Now you see, we have simplified our concept. So this molecular scissor, what does it has? It has 1 single RNA, it has a 1 single guide RNA, because the tracer RNA and the CRISPR RNA have been joined by this linker artificially, and it has only 1 protein it needs to cleave, because the Cas9 has the nuclease activity. It has 2 domains and it actually has the nuclease activity. So I am just simplifying the whole set. So that is why the type 2 Cas system, that is the CRISPR-Cas9 system, has been most accepted, and it was on this that the majority of genome editing studies have been developed. So if we look into this system, see, this is the single guide RNA. The single guide RNA actually recognizes the target sequence along with the PAM, and then it helps in guiding the Cas9 to that area. The Cas9 will now induce a double-stranded break in the 2 DNA. What happens? We started with this, that whenever we are using a gene editing tool like this, it so happens that it uses 2 DNA repair machinery, non-homologous end joining, and homology dependent repair. So in case of non-homologous end joining, that is a bit error prone, but this is perfect, because if we have a donor DNA and there is a cleavage, then with the help of the donor DNA template, the DNA can be, the new DNA can be synthesized, and gene editing is thus done. So this is the modification done from the original CRISPR-Cas9 to a modified CRISPR-Cas9. The bacterial system consists of separate tracer RNA, separate CRISPR RNA, and a Cas9 protein. But our modified system consists of a single guide RNA that actually is nothing but the tracer RNA and the CRISPR RNA joined by a linker and the Cas protein. Now, coming to the categories of interference. So this is a beautiful picture. Now, as you can see, there are 5 type 1, type 3, which are very common, type 2 and type 5 interferences have been shown here. Now concentrate on type 1. In case of type 1 interference, as you can see, it is carried out by the cascade, cascade and Cas3. So this is the cascade. As you can see, this is the cascade and this is Cas3 separate. Okay, so this is the group of enzymes that are joined together. Now, cascade, as you can see, is a large complex containing the CRISPR RNA. You can see the CRISPR RNA. It's a large complex containing the CRISPR RNA bound at either end by Cas5 and Cas6, multiple Cas7 subunits along the CRISPR RNA, and a large subunit and sometimes small subunits. So we have a large subunit and sometimes we have certain small subunits also. So this is the total type 1 CRISPR-Cas system. Here a picture of type 1E is shown here. The large subunit recognizes the PAM in foreign DNA. So as you can see, the large subunit CSE1 actually recognizes the PAM in the foreign DNA, and you can see the backbone, that is the Cas7, and the RNA is actually annealing with, after recognition of the PAM, is actually annealing with the DNA of the invading genome, resulting in an R loop and makes a nick. It then Cas3, as you can see, that there is a nick made, and once the nick has been made, what happens? The Cas3 is recruited. Now the Cas3 is recruited to this complex, and then the Cas3 progressively translocates along the displaced strand and progressively degrades it. So the recognition is done by this cascade, and finally, once the recognition has been done, the annealing has been done, then Cas3 is recruited. Now let us look to the type 3 system. While I had showed another picture of type 3 system, I had said that in case of type 3 system can target both the single-stranded DNA and the RNA that is being synthesized from the DNA, because the virus will also take no time and wait, and will not wait. The viral genome will also not wait that when a CRISPR-Cas RNA system will come and attack me. So the moment it enters, it goes into work. It starts utilizing the bacterial translation machinery, the transcription machinery first. It starts utilizing the bacterial transcription machinery to start transcribing RNA copy of its DNA. So this is the type 3 system. The type 3 system contains either CSM or CMR complexes, as you can see them, complexes. And in this picture, the type 3A has been shown. The CRISPR RNA is bound at either end by the CSM5. So you can see by the CSM5 and the CSM4, which has homology to Cas6. There's Cas6 and Cas5 of the type 1 system. So it has homology to the Cas6 and Cas5 of the type 1 system. Okay? Now the CMF4 forms the backbone of the complex. So this is the backbone of the complex. As you can see, the CSM3 CMR4 forms the backbone of the complex, while Cas10 serves as the large subunit, and CSM2 or CMR5 are the small subunits. So these are the variety of names. Now, as I said, this complex can target both DNA and RNA. So it can target the DNA as well as the RNA. Cas10 catalytically cleaves the target DNA, while the backbone subunit, as you can see, the backbone subunit catalyzes cleavage of the target RNA at every 6th base. As you can see, they are being cleaved. So the backbone is cleaving the target RNA at every 6th base, which is unpaired with the CRISPR RNA. As you can see, the CRISPR RNA is not perfectly paired. So there is a areas where the CRISPR RNA is not paired. So it is in these backbones, as you can see from the picture. If you just magnify it and see it for yourself, you'll be able to see that it is in these backbone areas that the cleavages are being introduced. Now, as I had said, that PAM is present in case of type 1 and type 2 system to identify the protospacer. In case of type 3 system, I had said earlier that there are mismatches that needs to be identified that needs to be present to identify the target sequence, and I had talked about the 5' handle and the 3' handle. So these are the handles. You can see the CRISPR RNA has not perfectly paired with the target protospacer. It has not paired with the target RNA. It has not paired with the target protospacer. There is a mismatch. It is this mismatch which tells the CRISPR-Cas complex that indeed this is a foreign DNA. Had this been completely paired, now let us assume that had this been completely paired, then the system would have read it as its own DNA. Since we are having this kind of handles, so there are mismatches here, these handles are telling them that indeed this is the foreign DNA, this is not the self DNA. And finally, coming to type 2 interference by type 2 systems. So this is Cas9. I just talked that Cas9 has two lobes, a helical lobe and a nucleus lobe. In type in type 2 system, Cas9 forms the effector complex with the CRISPR RNA and the tracer RNA. As you can see, this is the tracer RNA and this is the CRISPR RNA. Cas9 is composed of the nucleus lobe and the alpha helical lobe. The nucleus lobe contains both the HNH and the roof C-like nucleus domains as well as the PAM interacting domain. You can see the nucleus lobe, it can interact with the PAM. It also has the two nucleus domains. The 3' hairpins of the tracer bind the nucleus lobe. You can see the 3' hairpins of the tracer is binding with the nucleus lobe, while the stem loop and the spacer line channels between the, so the stem loop and the spacer RNA channels in between here, the two lobes, binding to a matching PAM adjacent target. So when they bind to a matching PAM adjacent target, causes the HNS nucleus domain to move into a position. So it moves into a position to cleave the annealed strand, while the displaced strand is fed into the roof C site for cleavage. So the strand which has been annealed is cleaved by the HNH, while the roof C makes a point to cleave the strand which is displaced. And finally, we see here the type 5 interference. It is done by the CPF1 effector protein in type 5A systems. It is the best characterized one. It binds to the CRISPR RNA alone, and mind it binds as a dimer. That's what has been suggested. It binds as a dimer. The structure of CPF1 is not yet known, but it contains an active groove-like nucleus domain for target cleavage. The CPF1 recognizes a PAM and makes two staggered cases. It recognizes the PAM and makes two staggered cuts in a matching sequence. It has been proposed, as I was saying, it has been proposed that the CPF acts as a dimer with each monomer providing the roof C site, although there might be unidentified. So this is a matter of much work needed in this sector, although there might be unidentified nucleus domains present in this context. So this was in detail about the Cas machinery. In generalized, there is a basic protocol, and then based on the different category of Cas proteins involved, we have different types of CRISPR systems. And based on the different proteins involved, we have a variety of interference. And of this different interference, if you look into this picture, in the first go, each of one will select this because it involves only one, as I said, it involves only one protein. And if somehow this tracer and the CRISPR RNA can be linked, then it involves one single guide RNA. So this is the basic concept. Now we are moving on to the applied part, CRISPR-Cas and genome editing. So this is a very schematic picture. So this is the Cas9 and this is the guide RNA, the single guide RNA. They recognize the PAM. So they search for the PAM and the target sequence. The guide RNA recognizes the target sequence. And then once the target sequence is recognized and the guide RNA anneals to it, the nuclease domains of the Cas9 are activated and the gene is cleaved. So these are the two basic mechanisms. As I said again, there's a similar picture here. So these type 1, this is the type 2 mechanism, this is the type 1 mechanism. The type 2 mechanism is the one that is most worked out, and you can see a particular nomenclature here, which is again I have not yet said till now, the dCas9. So there's a difference between Cas9 and the dCas9. I'll come to that a bit later. And how are these complexes introduced into the system? For different formats of gene editing, the whole DNA format can be introduced. If the whole DNA format is introduced, then it will accordingly initiate or necessitate the various paths. So it will the sgRNA will be produced, the Cas proteins will be produced, the sgRNA, so the ribonucleoprotein complex will be produced. And the CRISPR RNA, that the single guide RNA will now guide the CRISPR, the Cas protein to the target site, and Cas will initiate the cleavage of the target site. The simple guide RNA, in the RNA format, they can be introduced into the cytoplasm, or they can be introduced into the cytoplasm in the Cas9 SG format. In whatever manner they introduced into the cytoplasm, as I said, they require two DNA repair mechanisms to work in their favor. Not two, the main one is the HDR pathway. They actually involve two DNA repair mechanisms, the non-homologous end joining and the homologous and the homology dependent repair. So that is the two pathways. So we have discussed. And as I said, in the non-homologous end joining, what happens? There is a problem. So as you can see, the Cas9 is being guided by the single guide RNA to a target site. DNA cleavage, the ends are ligated in the non-homologous end joining, but there may be insertion, deletion. Together they are known as indel mutations. This is an error-prone one. But there are applications to it. Presently a lot of gene targeting studies are being done by it, and again we are seeing the word dCas9. I'll come to that. And then we have this mode of repair mechanism where we can we can enter a gene of our interest. So if there is a cleavage, double-stranded cleavage being inserted by the CRISPR-Cas9 system, then, and if a donor DNA is supplied, then the donor DNA will act as the template and the gaps will be repaired by using this red sequence as the template, and as a result, our target sequence of interest will be knocked in, will be inserted in. And these are the various fields. These are the major application areas of CRISPR-Cas-based technology. They are involved in gene editing, gene regulation, epigenome editing, chromatin imaging. I hope I'll be able to show you a small video. So there you will see how they are also involved in epigenetic editing. Then there is RNA targeting, chromatin topology. So it's a whole sphere of a multi-domain area where the CRISPR-Cas9 technology can be used. Now coming to the word, I said that yes, there is a difference between Cas9 and dCas9. The D word stands for dead. Fact is that when we introduce Cas9 sgRNA in this format, we have the Cas9 and the sgRNA, that's a single guide RNA. Cas9 is having its nucleus activity intact. So it will go and cleave the target DNA. But when we want to modulate the expression of a gene, we don't want to cleave the gene. We just want to silence the gene, or we want to express the gene. There's no point in cleaving the DNA segment. So what is done? So this dCas9, it's an alternative product where the nuclease domain of Cas9 is inactivated. Okay, so the nucleus domain of Cas9 is inactivated. In most cases, as you can see, mutations in the nucleus domain is commonly used in case of Streptococcus pyogenes Cas9, which results in the Cas9 variant referred to as dead Cas9 or dCas9, which still is able to bind to the target sequences by virtue of having this sgRNA. Actually, the sgRNA helps in binding to the target sequences. And the dCas9 binds, but the dCas9 is now not able to cleave the target sequences. So how can this mechanism be used? So here what happens, the sgRNA has been synthesized in such a manner so that it targets a promoter element. So the sgRNA targets the promoter element and it has its dCas9 component. As I said, since the dCas9 component is present and it binds with the sgRNA, it cannot cleave the DNA, but being present in the promoter element itself, it blocks RNA polymerase from binding to the promoter. So in other words, it actually represses transcription. So somehow it has repressed the transcription of the gene for which this promoter is the controlling element. Now, the other way around, if you want to enhance the expression of a gene, in such cases, as you can see, there is a transcription factor that has been recruited and added to the Cas9, dCas9, and this is usually the omega segment of RNA polymerases. For cases where there has been experiments done on expressing the transcription, enhancing the transcription level. So the omega segment is a part of the RNA polymerase. So the omega segment has been added to this the halo, the violet halo is actually the dCas9 complex. So the omega segment has been added to the dCas9 complex, and as a result of addition of the omega segment and the and the guide RNA has been targeted in such a manner so that it sits just upstream of the promoter, and this omega segments now help in recruiting the RNA polymerase, thereby increasing the transcription level manyfold. So these are dCas9 is a modified version of Cas9 when we are thinking of not actually cleaving the DNA, but modulating the expression level of a gene, dCas9 is used. This is an example of how the CRISPR-Cas, it depends on what you want to do with your genome. If you want to cleave your genome and insert a gene in, that's knock in, you insert a gene. So, or what you can do, knock out, you remove a gene. So it depends on what you want to do. That based on that you choose your Cas9. So if it's simple to insert a gene or knock out a gene, you choose Cas9 because it is having its nucleus activity intact. But in cases where you do not want to insert or edit out a gene, but you want to change the expression level of the gene, then you have to involve dCas9. So that's the basic difference about what you want to do with the gene in hand, the gene you have in your hand, what you want to do. Now, this is an example how genome engineering has been done in plants. And as you can see, this is the sgRNA directs the sp-Cas9 protein. This is a modified Cas9 protein to bind to the genomic DNA with a 20 nucleotide sequence and further induces it to introduce a double-stranded break. This double-stranded break, then as we said again and again, can undergo two repair mechanisms. Now, if we choose this homology-dependent repair mechanisms where we supply our donor DNAs, so we get the inserts. So we have inserted a gene of our choice in the blunt ends. So this is the timeline of a number of fruits that were in fact been first applied, where this genome editing process was first applied. As you can see, it started from 2014 and is continuing into up to 2018, and this process is continuing with. So there are a number of fruits like tomato, then we have cucumber, then melon, then banana, ground cherry, and so on. This is another example of how to target antibiotic resistance gene. If the antibiotic resistance gene is known to us, we sequence our sgRNA in such a manner so that it can target that antibiotic resistance gene. So we have our sgRNA, we have our Cas9. Again, we are using Cas9, not dCas9. So our aim is to cleave the antibiotic gene. And this genome, I mean to say the sgRNA Cas9 is then inserted into a phage DNA and then into phage meat, and the phage meat infects the bacterial cell. It inserts our sgRNA and Cas9. The sgRNA targets, I mean to say guides the Cas9 to the targeted antibiotic gene, and then Cas9 then efficiently cleaves off that antibiotic resistance gene. So this is how the gene has been cleaved off, and this is another simple system where in cancer cells this can also be used, where the sgRNA and the Cas9 are introduced. See, for example, here if we see this example, this is a normal cell. In normal cells, the LAC-I gene is present. The LAC-I gene represses the LAC operon. As a result, the oncosuppressor is inactivated. But in presence of the Cas9 sgRNA system, the LAC-I gene is inactivated. Since the LAC-I gene is inactivated, in presence of the LAC operon, the oncosuppressor is activated. So these are modifications. There's a variety of modifications that can be done. Some has made success. Another way of targeting cancer cells is that simply you target the oncogene. If the oncogene is known to us, simply target the oncogene and the oncogene is inactivated. CRISPR-Cas9 and sgRNA complex is packaged into virus or non-viral systems such as liquid nanoparticles to deliver it into target sites such as liver. And in scheme of ex vivo genome editing, adult stem cells such as the hematopoietic stem cells or the induced pluripotent stem cells can be targeted for treatment. They can be taken out, cell correction done by the CRISPR-Cas9 system and may be reintroduced into the patients, or the somatic cells from skin or can be reprogrammed in presence of the induced pluripotent stem cells. CRISPR-Cas9 editing can be done, differentiation introduced, and can be further introduced into the cell, into the body. So these are a variety of strategies that are opening up. And before ending into my PowerPoint, since we are sitting in COVID times and we are onto a digital platform having a webinar, the CRISPR-Cas9 system had been used as a rapid assay to detect the viral load. What happens actually is that once the viral RNA is, extracted and then subjected to isothermal amplification by primers that target the E and the N2 gene of the SARS-CoV. If the Cas12a sgRNA complex binds to the target sequences, the Cas12a activity gets activated, and as a result, it brings about indiscriminate cleavage of the RNA. Now, the RNA has a fluorescent tag and a biotin tag. Now, in the lateral strip flow, the biotin tag, since the probes are tagged, it is a biotin tag. Since the probes are tagged with streptavidin, the lateral flow strip first captures the streptavidin portion of the probe via the biotin. So the probe is actually the streptavidin. It is captured via the biotin. So it's captured via the biotin, and the fluorescent probe will continue to flow through the system and will be captured by an antibody further along the stream. So this is how the CRISPR-Cas9 system has been used. In this case, this is another methodology. I'm not getting into this one. Here the CRISPR-Cas9 methodology has been used. I just gave one example. This talks about the CRISPR-Cas12. So the CRISPR-Cas strategy has been used to identify the viral load in infected organisms. That is how the rapid tests are being done. And when we use something like this, when we use to cut RNA, to insert a segment of gene, to knock out a segment of a gene, ethical issues tend to come off. So, however, when we are talking about replacing endogenous disease-causing genes, there is a moderate risk. But if you think of germline modification, that becomes very high and unpredictable. In case of animal models for research to study diseases or developmental process by mutation or silencing genes, basically for silencing purposes, there is no risk, because ultimately you're not cutting off the gene, or you're not inserting any gene. You're just modulating the gene. So you're just doing tweaking with the epigenetic system. So that is not much of a risk. In case of plants, improving crops, introducing disease resistance, controlling harmful invasive species. Now, the risk level is fairly high. So this was a debatable thing, as I said. Research is going on. This is an active research sphere. So more, so documental evidences need to come up into this area to make this totally risk-free zone. So as you can see, the potential of CRISPR application is huge. There is cancer and tumor biology, plant biology, engineered ecosystems, somatic genome editing. So it's huge. But along with it comes the negative side. There might be long-term effects. As you can see, the papers are from 2013, 16, 18. So we do not know. After we have introduced a knocking gene and introduced a new crop, we have introduced improved variety of crop, fruit. But after 10 years, what's going to happen to that, we don't know. Then there's the ethical breach, there is safety, there is regulation. So these things needs to be taken into care of. So these are the concerns basically. And finally, I would like to end here. So this is the DNA that is worried, because the CRISPR-Cas is there. It has the sgRNA bearing the sequence of this DNA. And since the sgRNA has the sequence of the DNA, the CRISPR-Cas is very confident and is saying that, yes, wherever you are, I'll be able to find you. And if it's the Cas9, then I'll cleave you. So I thank you all for listening to this long lecture with such patience.
Malabika, there are few questions. Would you like
আচ্ছা আগে কি questions নিয়ে নেব?
এটা তোর ইচ্ছা যে আগে কোশ্চেন নিবি না আগে ভিডিওটা দেখিয়ে নিবি?
ভিডিওটা 9 মিনিট এর, will time permit that? তাহলে ভিডিও দেখাবো না, তুমি বলো।
এখন টাইমে ওটা মানে ওরকম কিছু না, মানে 5-7 মিনিট চলে গেলে কোনো অসুবিধা নেই। If you think the video will make the understanding a little bit
তাহলে ভিডিওটা দেখাই, ভিডিওটা আমি ওদের
ওকে দেন ভিডিওটা দেখানোর পরেই আমরা মুভ করব।
ওকে, দাঁড়াও শেয়ার করছি, শেয়ার স্ক্রিন। Is the video being showed? ভিডিও দেখা যাচ্ছে?
হ্যাঁ দেখা যাচ্ছে, সাউন্ড ছাড়া।
ঠিক আছে, okay, fine. So it's a video and you just see how it goes. I'll be putting in some inputs where it is needed. So as it is saying that this CRISPR-Cas9 is now being used in a huge area. So it's the broadening of the area. Now it's talking about the applications. So it's, as you can see, as I said, that in each category of types, a lot of modifications of the CRISPR-Cas9 system is done. Now I was saying that CRISPR-Cas9 can be used, CRISPR-D-Cas9 rather, can be used for genetic modifications. Now let us see. So this is that sgRNA, and this is the target sequence, and this is our Cas9 endonuclease, as I was telling you again and again. And it has a nuclease domain, so it can cleave the two DNA, and it actually identifies the target sequence by the PAM motif. This is a very famous PAM sequence from Streptococcus pyogenes. Now, when this kind of cuts are obtained, blunt DNA cuts are obtained, the cut can be done, repaired by two mechanisms. Is the NH-EJ pathway, where it's the joints are ligated, but there might be insertions or deletion mutations. So they can disrupt gene expression or gene function. But if there is a HTR, in the HTR repair pathway, there is a template. This is the left homology arm, this is the right homology arm, this is the desired sequence, and the desired sequence is then filled out. Now coming to dCas9. These are the various CRISPR endonucleases that are being shown here, various CRISPR endonucleases that are used in multiple systems, depending on what I want to do, whether I want to regulate, whether I want to cleave, whether I want to go for epigenetic modifications, depending on that, what I want to do. So from this, you choose your choice. Now, the problem is that edits are permanent. We have to keep this in mind. They can be toxic to cells. So there's an ethical consideration. Coming to dCas9, as I was talking about. So in this case, it is losing its power of cleaving the DNA. So as you can see in this picture, so this is a gene. So upstream in the promoter element, the dCas9 CRISPR complex has the sgRNA has recognized the promoter element. And sitting here, it can repress the gene, or as I told, it can increase the gene expression if a transcription factor, a part of the omega component of the RNA polymerase is added to it. So it can be either or. Now, normally, initially, gene silencing were done by these three basic methods, but they become difficult when you to do a bit elaborate processes. Now, dCas9 systems are very good in activation and these are very good activation and repression systems. And as you can see, this is a very famous model used. So this is the dCas9 SAM system. And in this is the gene of interest. And there are two molecules added to this Cas9 molecule, VP64 and P65. So if this is the gene of interest, they act as transcriptional activator, thereby bringing in activation of the gene expression level. Similarly, in case of this is actually in case of HIV, H1N1, it has been shown some successful results. So this is the picture of those HIV viruses. Similarly, there is another system known as a Krebs system. This is a repressor. So by recruiting this repressor with the dCas9, the repression of the gene of interest can be done. Previously, we knew that in cases of gene silencing, the different non-coding RNAs are actually obtained from a variety of degraded forms of RNAs. But right now, and the Cas9 Krebs system can target both the non-coding RNAs, the microRNAs, the antisense transcripts, and the nuclear localized RNAs. Now comes to the concept of epigenetic editing. In case of epigenetic regulation, so there are two genes. The two systems. We can silence a gene or we can activate a gene. Now, I think so you all are aware that there's a phenomenon known as methylation and there's a phenomenon known as acetylation. When histone acetylation occurs, the DNA becomes accessible. It's open form. When histone deacetylation and DNA methylation occurs, the DNA becomes inaccessible. So that is how the epigenetic control is done. So this is a second level of gene expression. This is heritable, and to some extent, this is reversible. Now, this particular mechanism of epigenetic regulation can be used in a variety of biomolecular applications and in a variety of diseased conditions also. Initially, the other strategies were laborious, unreliable, expensive. But with this system where we can add molecules to the Cas9, we can have an activator domain or a repressor domain, or we can add histone modifiers to the Cas9. We can add methylation modifiers. So these modifiers actually will modify the the conformation of the DNA along the histone. So this is one modifier, that acetylase histone. And as you can understand, if histone is acetylated, the target DNA will open up. As a result of it, we'll see expression of the gene. This is methylase. The target DNA will be methylated, resulting in silencing of the gene. So by modifying these methylations and acetylations, the dCas9 CRISPR system will thus provide much more implications in cell development, cell differentiation, and diseases. dCas9 fused to DNA manipulating effect enables the study of methylation-related silencing and development of therapy combines. So, sorry. This is another where the TET1 molecule is added. So dCas9 TET1, it is added, which helps in methylation of the CpG islands. You people are aware that the CpG islands are present in plenty of promoter elements. And if methylation is done in the CpG islands, the gene is silenced. And one success that has been achieved by methylation of the CpG islands is in the case of BRCA1, that is responsible for breast cancer. This is another modifier. So this is how a variety of modifiers, depending on what we want to do, can be added to the dCas9 system, and accordingly we can modify, or accordingly we can do what we want. So you can see the variety of dCas9 systems, what we want to do. So with this is what I wanted to show, Papiyadi. So with this, I think so I'll stop sharing.
Okay.
Okay.
Okay. So before taking question, I must thank Malabika a lot. I must say that this CRISPR-Cas technology, to understand the fundamental science for this technology, is very crucial, and it is quite tough also. There are so many tidbits for this, and I have really doubt that those people or researchers who are using this technology, they hardly really understand the basic science beside this. Beside the ZFN and TALEN, which are the techniques that are still used in genome editing, CRISPR-Cas is the most violent one. But it is really, really tough without a thorough understanding and high-level grip in molecular biology. It is impossible to elaborate the system to the students. I know there are students from various levels, and since it is an advancement program, so I thought that it is better to incorporate varieties or different levels to give you the essence. This will be pretty hard for the beginners, but still, after this nice explanation, though those people who are junior may have a little knowledge of the tidbit of this kind of molecular technique. Those students who are in PG, I think they have enjoyed a bit more, and they will have much understanding about the basic science after Malabika's elaboration. And I must appreciate this is such a tough technology. I mean, the technology is easy, that Malabika has described in the video. But the basic science for the technology is very hard. Is very hard. When I also gone through the paper, it required 2 to 3 days to understand the technology.
Yes, that's true.
So Malabika has started from the very basic, and it is really it is a really overdose for the beginners. But since it is, I am repeating, যেহেতু এটা একটা স্টুডেন্ট অ্যাডভান্সমেন্ট প্রোগ্রাম, তাই জন্য অ্যাডভান্স নলেজের একটা ভিউ বা গ্লিমস তোমাদের থাকা উচিত। এবং যাদের, দেখো আমরা চেষ্টা করেছি, উই হ্যাভ অ্যাকচুয়ালি tried to synchronize different aspects, কারণ এক একজনের এক এক দিকে ইন্টারেস্ট। Those who have interest in molecular biology and biotechnology, they will definitely enjoy this lecture. They would have definitely rather enjoyed the lecture, এবং সঙ্গে সঙ্গে কি হয়, আরো জানার ইচ্ছাটা বেরোয়। এবং এটা খুব সত্যি কথা যে এখন এই টেকনোলজি দিয়ে তুমি যা বলবে তোমাকে বানিয়ে লোকে দিয়ে দেবে। বেসিক্যালি ইউ হ্যাভ টু অ্যাপ্লাই দ্য টেকনোলজি, বাট উইদাউট knowing the basic science you will never enjoy the technology. আমি টেকনোলজি ব্যবহার করলাম, পিএইচডি করে বেরিয়ে গেলাম, কিন্তু টেকনোলজির ফান্ডামেন্টাল জিনিসটাই আমি জানলাম না। এটা কিন্তু সব জায়গায়, যেটা আমি তোমাদের বায়োস্ট্যাক নেওয়ার সময়ও বলেছি, যে কেন ফান্ডামেন্টাল জিনিসটা তোমাকে জানানো হচ্ছে, তুমি একটা সফটওয়্যার ইউজ করছো কিন্তু ইন্টারপ্রিট করতে গিয়ে দেখছো যে তুমি কিন্তু বুঝতে পারছো না, কারণ তুমি আসলে বোঝোনি যে আমি কিভাবে অ্যাপ্রোচটা নেব। That's why Malabika, very 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 nice elaboration. It's a really tough one, এবং সত্যি সবটা মানে ওদের it is a long process to make them understood, কিন্তু আমার মনে হয় ওরা অন্তত টেকনোলজির একটা বড় অংশ বুঝতে পেরেছে, ফান্ডামেন্টাল অংশটা। Anyway, now the question is yours. তুই কি নিজেই কোয়েশ্চেন গুলো নিবি না আমি পড়বো? কোয়েশ্চেন আনসার ক্যান ইউ সি?
I can see chat box এ আছে না, কোনটা দেখো বলো তো কোয়েশ্চেন?
Q&A আসছে।
হ্যাঁ দাঁড়াও দেখছি, সরাচ্ছি, দাঁড়াও এক সেকেন্ড ক্যান্সেল, লিভ মিটিং হয়ে যাচ্ছিল। Yes, I can see. Is Caspase 9 here? This is Otindra Korar. Ma'am, is Caspase taking part in Apoptosis? No. This Caspase 9 and this Cas9 are different. Now those who have studied apoptosis, they make this very big mistake. But Caspase 9 is a different sort of enzyme. And Caspase 9 is a much more enzyme, and over the evolution it is present in a lot of phylogenetic groups, but Cas9 is restricted to certain groups only. Up till it is we just
associated.
Yes, it's CRISPR associated. If you remember the full form, it is CRISPR associated protein. So those two are different. Okay, I hope I answered this question. Is it mandatory for the foreign DNA to have either PAM? Yes, it is mandatory for the foreign DNA to have PAM or a 5' 3' flanking mismatch or a PFS, as I said, protospacer flanking sequence, because how can see if a virus has decided that this should be a strategy of adaptive immunity, then the bacteria has to also make it sure that it is foolproof. If I make something expending energy, I need to make it foolproof. So this is a foolproof system. So there is a context on basis of which the target sequence is recognized. So the presence of PAM, as far as the work that has been done till now, this is still a developing field. So as far as the work done till now, the presence of PAM, the flanking mismatches and the protospacer flanking sequences are essential till date. Ma'am, I want to know, how does CRISPR, see, CRISPR-Cas system is something programmed. You cannot decide a viral genome as lethal or not lethal. It's a programmed sequence to identify the PAM, and if there is a PAM, if there is a mismatch, if there is a PFS, the protospacer of the target sequence will be recognized. So it's a programmed system. It is not a matter of choice, it's a matter of programming. So the CRISPR-Cas system in bacteria actually recognizes any foreign DNA that has a PAM, and in context to the PAM, it has this target sequence. Okay. So I think so I answered that. If they are not satisfied with their answer, Papia Di, you can send my mail to them. Then they can contact me, or you send me, I can write the answer.
Okay.
Acha, it's tough to remember all this information just in one class. I get that. After teaching for almost 15 years, this topic was tough for me, and I took some time from Papia Di. She wanted to give me another day, and I said, no, you give me this day. So it's a tough one. I agree to that. It's tough to remember all these things.
It is
I
quite
very much
tough for
agree.
them. Even that's why আমি ওটা বললাম যে আমি যখন নিজে জিনিসটা পড়েছিলাম তখনও এটা আমার 3 দিন 4 দিন লেগে গেছিল পুরো
এক্স্যাক্টলি ওটা লাগছে।
বুঝতে।
and মানে আমি একটু বলি বায়োটেকনোলজিস্টরা কিন্তু
you will be the right person at least কিছুটা ওদের বোঝানো সবটা সম্ভব
আশা করি বুঝেছে। আমি একটা জিনিস বলি যে it's just like cooking biryani. When somebody gives you a recipe of biryani, it becomes tough when you read the recipe and then you start cooking. Okay. Now, while going through the recipe, each recipe has a function. Now, what the biotechnologists do, they get the recipe and they cook the biryani. But each recipe, individual portion has a function. Usually the biotechnologists don't get into that. That is the classical part. So as I said, cooking biryani is tough. So it was something I cooked today. Then ma'am, for human,
some research scholar they use for their transgenic this CRISPR-Cas because we have biotech companies and you can simply order that I need this and this, and by CRISPR-Cas they prepare, we utilize it, but actually very few people really know the science background. But there is really if you know the background, it really means something different. আমার একজন টিচার হিসেবে মনে হয় যে আমাদের স্টুডেন্টদের মধ্যে এই ব্যাপারটা ইনকালকেট করার চেষ্টা করা উচিত যে বেসিক সায়েন্সটাকে একটু ভালোবেসে বোঝো। এবার আমার 230 জনের মধ্যে যদি দুজনেরও মনে হয় যে না এবার আমি সত্যি পেপারটা পড়ে বোঝার চেষ্টা করি, সেটাই আমাদের কিন্তু প্রোগ্রাম আউটকাম। I think that will be a real achievement. Even if 1% of them get interested and they just take the paper and go through it, that's enough. That's enough for us. Acha, next, so many questions are there. Sound শোনা যাচ্ছে না মানবিকা?
হ্যাঁ, শুনতে পাচ্ছো?
Yes. হ্যাঁ।
Ma'am, for human genome is chromatin neuronal associated CRISPR-Cas9 before incorporate remodeling. এই কোশ্চেনটাতেও অ্যাকচুয়ালি কি জানতে চেয়েছে সেটাই বুঝতে ইস ক্রোমাটিন
Which question? শোনা যাচ্ছে না, একটু জোরে বল।
মন্দিরা ঘোষ ও জানতে চেয়েছে যে কমপ্লেক্সেস অ্যাসোসিয়েটেড ক্রিসপার
Ma'am, for human genome editing is chromatin remodeling complexes are associated with CRISPR-Cas9 before recognition. No.
This is something you are procuring from the market. So when you want to see chromatin remodeling, you have to procure from the market a Cas9 CRISPR system which has already either the activator or the repressor tagged to it, or the histone acetylase or demethylase tagged to it as to what you want to do. So it is not it doesn't happen after recognition. Then this whole system you introduce into the cell you want to study. Okay, so that is done before. And
the next question actually cannot be answered within such a small time. Each individual application of CRISPR-Cas মালবিকার হয়েই আমি বলে দিচ্ছি, বিকজ ও 2.5 ঘন্টা যথেষ্ট বকেছে এবং গলার উপর চাপ পড়ে, এবং এই মলিকিউলার বায়োলজি টানা 2.5 ঘন্টা পড়ানো কিন্তু যথেষ্ট স্ট্রেনাস beside other simple lucid things. সেখানে কি করে ভিশনটাকে কারেকশন করছে, অর্থাৎ জিনোম এডিটিং এর সবকটা অ্যাপ্লিকেশন তাহলে আলাদা আলাদা করে তোমাকে বুঝতে হবে। ইট ইজ আ লং টার্ম প্রসেস। এটা আমি জানি না, ইটস আপ টু মালবিকা। আমি
না তুমি ঠিকই বলেছো, এগুলো
আমার মনে হয়
না ইন্টারফেস ঠিকই করেছো। তারপরে কিছু CRISPR-Cas তো ডেভেলপিং, অনেক জায়গাগুলো ওয়ার্কড আউটও না ঠিক করে, ইটস নট ইয়েট ওয়ার্কড আউট। What is the difference of the refer? Why it has to be in between the spacers? আচ্ছা, এখানে পারমিতা লিখেছে, কোথায় আছি, দেবপ্রিয়াতে আছি। How was the CRISPR used with vectors for knockdown or knockout genes? This is a different story. This is a process of knockout system which has to be explained to explain this one. দেবপ্রিয়া যেটা লিখেছে ও নকআউট জানতে চেয়েছে।
How the CRISPR-Cas can be used, এগুলো প্রত্যেকটাই হচ্ছে স্লাইডের মধ্যে ছিল সবকটাই মালবিকা, কিন্তু ইন্ডিভিজুয়াল স্লাইড ধরে তোমাদের পড়াতে হবে। তোমরা ওই অংশটা এক্স্যাক্টলি দেখো, বেসিক সায়েন্সটা তোমাকে বলা হলো, তারপরে এই ইন্ডিভিজুয়াল যে কোয়েরিটা সেটা দিয়ে ওই অংশটা তোমাকে দেখতে হবে। প্রত্যেকটাই ইন্ডিভিজুয়ালি বোঝাতে গেলে ইট উইল বি হাফ অ্যান আওয়ার।
হ্যাঁ মানে এগুলো ধরে ধরে
প্রত্যেকটাই আলাদা আলাদা ধরে ধরে বলতে হবে। সো বেসিক কোয়েশ্চেন গুলো নেই আমার মনে হয়,
হ্যাঁ
যেগুলো অ্যাপ্লিকেশন বেস সেগুলো তো এক একটাই এক একটা লেকচার।
Exactly. আচ্ছা এখানে একটা আছে, what is the significance of repeat sequence? Now let me tell you something. When initially it was discovered, there was repeats and there was spacers. তোমাদের আমি একটা শুরুতে দেখিয়েছিলাম, তোমরা কাট অ্যান্ড পেস্ট ট্রান্সপোজিশন নিজেরাও পড়েছো, যখনই একটা স্পেসারকে অ্যাকোয়ার করা হচ্ছে ইন বিটুইন আ রিপিট সাইট, তখনই দেয়ার ইজ আ টার্গেট সাইট ডুপ্লিকেশন। এই যে রিপিটটার মধ্যে তো স্পেসটা ঢুকছে, অ্যাজ এ রেজাল্ট কি হচ্ছে, রিপিটটারও ডুপ্লিকেশন হয়ে যাচ্ছে। সেই হিসেবে রিপিটস গুলোরও যতগুলো স্পেসার পাচ্ছি ততগুলো রিপিটস পাচ্ছি। দিস ইজ হাউ দ্য রিপিটস আর জেনারেটেড। ইম্পর্টেন্স অফ দ্য রিপিটস, স্পেসার যখন ক্রিসপার ম্যাচিওর আরএনএ গুলো দেখা যায়, দ্য CRISPR mature RNA contains portion of the repeats as well as the spacer sequences. তার মানে বোঝাই যাচ্ছে রিপিটসদের একটু ইম্পর্টেন্স আছে ফর প্যাম রেকগনিশন। ওই রিপিটস গুলো দে হেল্প ইন প্যাম রেকগনিশন, দে হেল্প ইন রিকগনাইজিং দ্য mismatches, 5' 3' flanking mismatches, and they help in recognizing the protospacer flanking sequences. এটা একটা বেসিক ছিল।
So the next question is your student probably.
কোনটা বলো তো? If sigma yes, SGR is present after PAM sequence without an artificial linker with genetic no see genetic engineering can occur, SGRNA করা হয়েছে with linker, উদ্দেশ্যটাই হচ্ছে আমি সিস্টেমটাকে ছোট করে নেব। আমি এখন তিনটে মলিকিউলকে নিয়ে যদি কাজ করতে যাই, আমার সেখানে তিনটে জায়গায় যদি দুটোতে চলে আসে, দুটো আরএনএ কে জয়েন করে আমি একটা লম্বা আরএনএ করে দিলাম আর একটা প্রোটিন, ইট ইজ দেয়ার টু মেক ইট সিম্পলার। মানে আগেকার প্রেসার কুকার ইউজ করি না কেন, কেন আমরা ফিউচারের প্রেসার কুকার ইউজ করি, মানে কি জিনিসগুলোকে সিম্পল করা, ওইটা হচ্ছে দ্যাট ইজ ইট। But SGRNA is present upstream of PAM sequence without an artificial linker, but there should be the tracer. Cas9 সিস্টেম ইউজ করতে গেলে কিন্তু ক্রিসপার আরএনএটাকে থাকতে হবে, ট্রেসারটাকে থাকতে হবে, Cas9 কে থাকতে হবে, আদারওয়াইজ ট্রেসার ক্রিসপার আরএনএ প্যাম সিকোয়েন্সকে প্রপারলি রিকগনাইজ করতে পারবে না। হ্যাঁ, লিঙ্কারটার উদ্দেশ্যটাই হচ্ছে দুজনকে একজন করে নিয়েছে। আচ্ছা ক্রিমিনালসের মধ্যে ঢুকছি না আমি।
তারপরে ম্যাম আমাদের রিপোর্ট অফ নন স্পেসিফিক টার্গেট,
yes অফ টার্গেটস হয়, অফ টার্গেটস হয় অনেক সময়, যদি প্যাম রিকগনিশন না হয়, যদি টার্গেট সিকোয়েন্স রিকগনিশন না হয়, তাহলে অন্যখানে গিয়ে যদি বসে যায় তাহলে অফ টার্গেট ক্লিভেজ হয়, দ্যাটস আ নোন থিং।
Ma'am, if someone wants to modulate human cell gene, then how can one recognize the specific PAM sequence of that gene? এটা তো স্টাডি করতে হবে।
এক্স্যাক্টলি, স্টাডি করতে হবে এবং সেই মতো বানাতে হবে,
সেটাই তো অর্ডার করতে হবে।
হ্যাঁ সেটাই অর্ডার করতে হবে আর কি।
হ্যাঁ না, অ্যাকচুয়ালি জিনোম সিকোয়েন্স করা সেটা ওরা বুঝতে পারছে না যে ওইটা টার্গেটটাকে ফিক্স করবে কি করে। যেহেতু জিনোম সিকোয়েন্স করা রয়েছে, তোমাকে আগে ওই জিনোম ব্লাস্টে গিয়ে তুমি যে জিনটাকে চাও সেটাকে আগে দেখতে হবে, তার আগে পিছে কি আছে দেখতে হবে, এখানে যদি থাকে তবেই সেটাকে টার্গেট করা যাবে, সবাইকে করা যাবে। মধুরিকা, এটা আমার মেয়ে, দেখ কি জিজ্ঞেস করেছে।
ম্যাম ইজ দ্য CRISPR জিনোম হেরিডিটারি? হ্যাঁ, ব্যাকটেরিয়ার মধ্যে থাকলে ব্যাকটেরিয়ার প্রজননীর মধ্যে যাবে, যে ব্যাকটেরিয়াগুলো ক্রিসপার লোকাস দেখাচ্ছে তো সেই ব্যাকটেরিয়াগুলোতে যাবে।
এটা ইনবিল্ট।
If it is then once a bacteria is embedded, the same virus cannot affect them ever. Yes, if it can acquire a spacer of that virus, I mean if it can acquire a protospacer, মানে প্রি স্পেসারটা যদি acquire করতে পারে তাহলে ওই ভাইরাসটাকে আর ইনফেক্ট করতে পারবে না। Is PAM for one specific gene? If yes, the specific PAM sequence for every gene that has been sequenced till now. সব জিন সিকোয়েন্সের প্যাম, মানে তাহলে তো দেখতে হবে সব জিনের এসজিআরএনএ তৈরি হয়েছে কিনা। মানে প্যামটা কি জিনটাকে আইডেন্টিফাই করানোর জন্য, মানে টার্গেটটাতে গিয়ে যাতে এসজিআরএনএটা লাগে ঠিক মতো। তাহলে সেই ক্ষেত্রে আমার জিন, আমি যখন জিন টার্গেট করবো, এটা বোধহয় একটু বোঝা দরকার, জিনের সিকোয়েন্সটা আমাদের জানা থাকতে হবে, ওটা তো টার্গেট করা হচ্ছে। আচ্ছা, if you are able to cause
প্রিয়া বা প্রিয় কি বলছে?
হ্যাঁ, দাঁড়াও, ফ্রগে এসছি, if you are able to use the metabolism of a cell like frogs, can you introduce the tardigrade genome?
If we are able to cause the metabolism of a cell like frogs,
এটা একটা হিউজ আনসার, মানে এটা
can we introduce the
tardigrade genome
into the host genome? Can we find a complementary base pair? How environmental factor affects the gene editing? অনেক বড় প্রশ্ন এটা। It is a big deal. It is a big deal. If we are able to একই প্রশ্ন দিয়েছে।
হ্যাঁ হ্যাঁ, দুটো হয়ে গেছে। How expensive is CRISPR?
সবই করতে পারি আন্ডার ট্রায়াল। করতে পারবে
ক্রিসপার সিস্টেম তো ল্যাবরেটরির বাইরে
বাইরে ইউজ হয়নি, সেটাই
নেচারে সেই মডিফাইড অ্যানিমালস গুলো কি চরিত্র হবে
এটা বোঝা মুশকিল। সেটাই বললাম যে তো করতে অনেক কিছুই পারি, কিন্তু সব তো আর করা হয়নি, চলছে। হাউ এক্সপেন্সিভ ইজ ক্রিসপার?
এটা ডিবেটেবল, কারণ তুমি কি সিস্টেম ইউজ করছো আর কি ক্রিসপার ইউজ করছো তার উপরে, বাট এক্সপেন্সিভ তো অবশ্যই।
কত পড়ছে?
প্রায় একটা রিঅ্যাকশনস করতে প্রায় লাখ 2-3 টাকা পড়ে।
হয়তো
মানে একটা প্রিপারেশন, একটা লাইন বানাতে লাখ 1.5-2 টাকা মিনিমাম। মানে একটা জিনোমেড না, একটা কোনো জিন এডিটিং করার জন্য যদি তুমি বা ট্রান্সজেনিক করতে চাও,
ফেলিয়ার থাকে।
ওই তো সবকিছুই মানে এগুলো ওই জন্যই খুব হাই ফান্ডা ব্যাপার আর কি। ত্রয়ী
আচ্ছা, ক্রিসপেস ইনজেক্ট করে
Cas9 আর Cas9 এর পার্থক্য আরেকবার বলে দিতে বলছি।
Cas9 টাতে হচ্ছে যেখানে আমার Cas9 এর এনজাইমটা যদি দেখি, there is a helicase lobe and there is a nuclease lobe. Nucleus lobe দুটো এনজাইম আছে, RUF-C and HNH, তারা ডিএনএ টাকে কাটে, these are nuclease. But if Cas9 হচ্ছে everything is kept constant, খালি এই নিউক্লিয়াস ডোমেন দুটোতে, যে HNH আর RUF-C ডোমেনে মিউটেশন ঢুকে এই নিউক্লিয়াস ডোমেনটাকে ইনঅ্যাক্টিভেট করে দেওয়া হয়, যাতে যখন এরপরে আমি এটাকে ইউজ করব ও কোনো ডিএনএ কে আর ক্লিভ করতে পারবে না, ওই জন্য ডেড Cas9 বলে, এটা বলা হয়েছে।
সোমদেব is in rapid generation of cellular and animal models the application of CRISPR-Cas9 used? হ্যাঁ, করা হচ্ছে তো, অ্যানিমাল মডেলেই তো করা হচ্ছে। ও কি প্রশ্নটা এটাই করতে চেয়েছে?
না, র‍্যাপিড জেনারেশন বলতে বোঝা যাচ্ছে না ও কি প্রচুর প্রোজেনি বেরোবে, আমি বুঝতে পারছি না কোশ্চেন।
র‍্যাপিড জেনারেশন মানে ও মনে হয় ট্রান্সজেনিক অ্যানিমাল তৈরির জন্য বলছে। হ্যাঁ, ট্রান্সজেনিকের জন্য CRISPR-Cas ব্যবহার করা হচ্ছে তো, পুরো এক্সপেরিমেন্টাল, সবই কিন্তু এক্সপেরিমেন্টাল। মনে হচ্ছে সেরকম ওই প্রশ্নটা করতে চাইছে, যে মানে ট্রান্সজেনিক সেল বা ট্রান্সজেনিক অ্যানিমাল মডেল বানাবার জন্য CRISPR-Cas9 ব্যবহার করা হয় কিনা আমি জানি না, if I can get it properly, মনে হচ্ছে সেটাই বলতে চাইছে। মালবিকা, you can try to understand.
না, তুমি যেটা বলেছো আমার মনে হয় সেটাই তো, নকআউট তো হচ্ছেই। আচ্ছা ম্যাম, is it to any selection marker or fluorescent marker
Possible to introduce any selection marker?
for what? মানে কেন ইউজ করব? এখানে তো প্রথম প্রশ্ন আছে আমি ইউজ করব কেন, যদি আমি একটা জিনকেই টার্গেট করার কথা ভাবি।
হ্যাঁ,
এটা তো
এটা কাজ বেসড, কখন তুমি কি কাজ করবে এই অ্যাপ্লিকেশনটা পুরো বুঝে নেওয়া তোমাদের পক্ষে সম্ভব না। তোমাদের এখানে যেটা টার্গেট করা হয়েছে যে সিস্টেমটা একটা কি অদ্ভুত সিস্টেম, যেগুলোকে ইউটিলাইজ করা হচ্ছে, এবং সিস্টেমের বিভিন্ন অ্যাসপেক্ট কাজ অনুযায়ী কিন্তু সব বদলে বদলে যায়। এখানে বিশাল বড় ফিল্ড CRISPR-Cas, ম্যাডাম অলরেডি দেখিয়েছে 8-10টা ইউজ দেখিয়েছে, এবার এই প্রত্যেকটা ইউজের আবার অনেক ভ্যারাইটি আছে, এটা বিশাল বড় সিস্টেম। এটা আবার কি CRISPR কি রিভার্স করতে পারে এজিং? এটা কি সোমদেবী এটা করবে, এসএনসি অদিতির কলেজের সোমদেব এটা নিয়ে ভবিষ্যতে রিসার্চ করবি, এজিং
রিসার্চ করতে চাইছে, একটু জানতে চাইছে।
যে জিন, ওই যে অদিতি আছে, মানে যে জিনগুলো এজিং করে সেগুলোকে তো অনেকটাই ফাইন্ড আউট করা হয়েছে, যে জিনগুলোর এক্সপ্রেশন হলে এজিং হয় বা এক্সপ্রেশন বন্ধ হয়ে গেলে এজিং হয়, তুই সেগুলোকে টার্গেট করে নক আউট কর বা কিছু একটা। এটা আমরা উই আর লুকিং ফরওয়ার্ড, এভরিথিং ক্যান বি ডান, দ্যাট ইজ দ্য থিং। ইনকুইজিটিভনেসটাই সবচেয়ে বড় কথা। কিভাবে তুমি ব্যবহার অনেক কিছুতেই করা যায়, এটা দারুণ টেকনোলজি, সেই জন্যই এত হই হই। ট্যালেন্টকে ছাড়িয়ে এখন সবাই CRISPR-Cas9 নিয়ে। আচ্ছা এসজি, since this program is an collaborative program, so Malabika, Sujayda want to talk to you.
Malabika, firstly I want to congratulate you for this nice narrative and presentations of such a complicated topic within a long 2-hour span with lot of
within just 2
detailing.
days.
Within just 2
I
days,
just requested that you explain this
all necessary detailing, and it is whatever I have heard a few lectures on CRISPR-Cas9, but honestly your lecture is probably the most lucid one, whatever that can be done at that level, because it's a really complicated technology. And as you know that presently even the scientists do not do their own experiment with CRISPR-Cas9. They are just ordered this to the biotechnology firm, and there are several US-based or Japanese biotechnology companies, those who are actually manufacturing different types of laboratory animals breed using CRISPR-Cas9 depending on your order and requirement. So that is a big technology, but still it's very, very complicated. So many things, so many details are there. But your students, this for you, your Malabika ma'am entirely has represented this lecture in such a lucid way and tried to make a pictorial and videographic presentation is really appreciable. And Malabika is one of the best molecular biology teacher among all the college teachers. Really, I am associated with the undergraduate board of studies, and I know that that Malabika is one of the best. There are several other good teachers, but Malabika is one of the best teachers in molecular biology area in the college level. So I should congratulate once again Malabika, and I am thankful for her consent to deliver such a beautiful lectures within a short span of time. Thank you, Malabika.
Sujata, coming this from you is huge. মানে coming this from you that তুমি CRISPR Cas-টা এতটা মানে আমি simplified করতে পেরেছি, this is huge coming from both of you.
It's
I
true.
look up to
It's
both
true.
of you. I look up to both of you, so this is really huge. মানে আমি যেটুকু খেটেছি তার যেটুকু প্রাইজ তোমরা দুজন যা বলে have got it.
No, it's true because it's I have gone through some of the lectures, not several lectures on the CRISPR-Cas9, but what happened, you know, that people those who are not involved in the teaching, the problem of teachers and the advantage of the teachers and the problems of the non-teacher scientists actually depicted from your or portrayed from your today's lecture. The teacher could realize what will be the wordings and what will be the representation by which you could nourish a young mind, but the researchers could not. That is true. So this is the first time I heard a lecture on CRISPR from a teacher. So I think that is the best whatever I heard in the last few days on the CRISPR. So once again, I am really thankful from the part of organizer and hope in near future we will have you as an organizer, an active member in organizing such another outreach program for the undergraduate and postgraduate students.
Thank you so
Malabika.
much. Thank you so much.
ওকে স্টুডেন্টস,
আচ্ছা,
তোমাদের অলরেডি চ্যাট বক্সে ফিডব্যাক ফর্ম আমি লিংক দিয়ে দিয়েছি। এটা তোমরা 24 ঘন্টার মধ্যে সাবমিট করবে। সেখানে ম্যাডামের দেওয়া 4টে অ্যাসাইনমেন্টও আছে, ভালো করে দেখে নাও। আমি একটা পেপার পোস্ট করছি, পুরোটা না বুঝলেও অন্তত কিছুটা পড়ে বুঝলে পরে আরো যত পড়বে তত এটা ক্লিয়ার হবে। এটা জাস্ট একটা ইনিশিয়াল গ্লিমস। সুতরাং একটা মেটেরিয়াল আমি পোস্ট করছি পেপার, সেইটার থেকে আশা করছি ম্যাডামের দেওয়া অ্যাসাইনমেন্টের কোয়েশ্চেন গুলো তোমরা করতে পারবে। ওকে, তো আর আগামীকালের ক্লাসটা একটু বলে রাখছি। I must declare for all of you that tomorrow's class will start from 5 p.m. Okay, tomorrow's class will sharply start from 5 p.m. So you try to join around 4:50. Within 5, but around 4:50 you try to join. And our yesterday's class that was not conducted will be probably conducted maybe on Sunday. That class যেখানে তোমাদের মলিকিউলার বায়োলজি টেকনিক্স গুলো দেখানো হবে, পুরো ভিডিওটাই সাপোর্ট করেনি, ওটাকে আমরা অন্যরকম ভাবে মডিফাই করে নিয়েছি। কিন্তু যেহেতু আজ এবং কালকের দুটো শিডিউল আলাদা ছিল, সেই জন্য আমরা এই ভিডিওটা প্রবাবলি আইদার শনিবার রবি, এই দুটোই তোমাদের খুব ইম্পর্টেন্ট। এখানে তোমাদের আমরা যে ক্যারিয়ার গাইডেন্স বলেছিলাম সব লেভেলের জন্য দেব, সেগুলো আজকে আমরা গ্যাদার করছি, কিছু ইনফরমেশন গ্যাদার করা বাকি আছে। That will be explained to you on Saturday. Saturday তোমাদের যদিও আমরা ফ্রাইডের মধ্যে শেষ করব বলেছিলাম, কিন্তু দুটো দিন আমাদের তোমাদের এক্সট্রা বলে দিতে হচ্ছে, বিকজ ক্যারিয়ার গাইডেন্সটা আমরা স্যাটারডে রাত্রেবেলা মানে 7টা থেকেই করব। ব্রততী লিখেছে ইয়েস, অ্যাকচুয়ালি আমরা 7টাই প্রেফার করছি, কালকে 5টা করা হচ্ছে বিকজ আওয়ার ইনস্টিটিউশনাল হেড, আওয়ার প্রিন্সিপাল ম্যাম will bring one senior teacher who was ex or retired professor of the University of Calcutta Department of Botany. He is very much eager to share some view regarding the bioweapons or bio war with you. That's why উনি অনেক বয়স্ক, 80-র উপর বয়স, উনাকে অ্যাকোমোডেট করার জন্য আমাদের কালকে 5টা থেকে করতে হচ্ছে। So please try to be there 5টার সময়, 5টা বা 5:15 এরকম হয়তো উনি শুরু করবেন। ওনার বলার পর then you have class of Dr. Indranil Saha on endocrinology, and শনিবার আমরা তোমাদের একাডেমিক সেশনের সাথে সাথে ক্যারিয়ার গাইডেন্স এর যতটা ডিটেইলস বিভিন্ন লেভেলে দেওয়া যায়, অর্থাৎ ইউজির পর হায়ার স্টাডিজ বা ইউজির পর যদি কেউ হায়ার স্টাডিজ এ না যায় তাদের আর কি কি অপরচুনিটিস আছে, কি কি কোর্সেস আছে, বা হায়ার স্টাডিজ এ নিলে কোথায় কোথায় তোমার এমএসসি পিএইচডি ইন্টিগ্রেটেড রয়েছে, তার জন্য কি এক্সাম, হোয়াট আর দ্য ওয়েবসাইটস, এন্ড আফটার এমএসসি অলসো কিছু প্রোফাইল তোমাদের দেখিয়ে দেওয়া হয়েছে। কাল সেদিন কনাদো ওয়াইল্ড লাইফের যে অংশগুলো আমি তোমাদের সাথে শেয়ার করে দেবো, যে ওয়াইল্ড লাইফ রিলেটেড জব গুলোতে তোমরা কিভাবে যেতে পারো। আদারওয়াইজ বাকিগুলো অলরেডি সুমন স্যার তোমাদের ফিশারি রিলেটেড সেই স্লাইড গুলো তোমাদের সাথে শেয়ার করা হবে। তার সাথে আরো যতটা সম্ভব আমাদের কাছে যতটুকু আমরা গ্যাদার করতে পারছি ইনফরমেশন সবটাই তোমাদের শনিবার শেয়ার করা হবে 7টার পর। দ্য ক্লাস উইল বি আফটার 7 PM। সানডে অলসো আমরা ভিডিওটা দেখালে 7টার পরই দেখাবো, সেটা স্যাটারডে জানাবো। শুধু কালকে ইউ হ্যাভ টু বেয়ার উইথ আস, প্লিজ ট্রাই টু বি from 5, যদিও তোমাদের অনেকেরই কলেজে ক্লাস থাকলে আমার কিছু করার নেই, দে ক্যান জয়েন লেটার, বাট কালকে আমাদের ক্লাসটা 5টাতেই করতে হবে। সো থ্যাঙ্কস স্টুডেন্টস, নাও ইউ ক্যান লিভ, ইউ হ্যাভ লট অফ কোয়েশ্চেন্স। আর কোনো কোয়েশ্চেন থাকলে তোমরা আমাদের মেল করবে বা আমাদের যে গ্রুপটা আছে টেলিগ্রাম গ্রুপে তোমরা জানাও। আমি মালবিকা ম্যামকে কিছু সেন্ড করতে পারি, আমি নিজে পারলে কিছু উত্তর দিয়ে দিতে পারি, উই উইল ট্রাই ফর ইট। ঠিক আছে, স্লোলি, হতে পারে যে এক্ষুনি সম্ভব হচ্ছে না, বাট সিন্স দ্য টেলিগ্রাম উইল বি দেয়ার, যতক্ষণ না তোমরা সার্টিফিকেট পাচ্ছ ন্যাচারালি সেখানে আমরা কিছু উত্তর করে দেবো। ওকে, সো ইউ মে লিভ স্টুডেন্টস, ইউ মে লিভ, এন্ড থ্যাঙ্কস মালবিকা।
হ্যাঁ, থ্যাংক ইউ তোমাকে।
আমরা মোটামুটি আজকে এন্ড করছি। ওকে,
বেরোচ্ছি, টাটা, বাই, ভালো থেকো।
থ্যাংক ইউ, গুড নাইট।
I'll call you next week. হ্যাঁ, I'll call you next week.
ওকে ওকে,
ঠিক আছে।
থ্যাংক ইউ স্টুডেন্টস, থ্যাংক ইউ টিচার্স। Students, I would like to request you to leave the meeting. We have a departmental meeting, so we are waiting, because if we remove you then you will not be able to join further. So I would like to request you to leave the meeting kindly.